A very good morning to all of you. First, let's do a voice and video check. If everything is fine, let me know. Wonderful. So, a lot of you are still asking. I'm almost okay. I'm fine. I hope nothing goes wrong now, but I'm good. Thank you for asking. All right. So let's begin with our next chapter now. Now we are done with the two of the biggest chapters, and that's why these two chapters have taken so much of time. Now we are moving on to the chapters which will not take so much of time. And the first such chapter is biotechnology. Now in case of biotechnology, again, first speaking from perspective of your prelims and mains examinations, this again is a chapter which will be important for both examinations, where we do see questions coming in prelims every year or in mains almost every year. So that's why, and again, the questions at mains level has been fairly easy, and in case of prelims examination, there have been certain ways in which they always have asked questions. Now when it comes to prelims, and I'm telling you upfront about this so that when you go through the chapter with me, you should always have this in your mind. There are two types of questions that biotechnology wise they ask. One has been a basic concept based question where they'll ask you the concepts based on biotechnology, and second, in biotechnology specifically, they have been asking a lot of word based questions. Now what I mean by word based questions are that there will be just certain words which will come in the news for one reason or the other. They just keep on picking these words and asking the meaning in just one line without explaining, without going into the details, the technicalities, just the definition. So that's why, apart from everything that we are doing, what also becomes important is that you always make sure that you understand what exactly has been there in the current affairs. And in current affairs, if you come across a new word that you don't know the meaning of, then you just pick that word, see the definition of the word, understand the definition, and that's it. And that will be enough for you to answer such word based questions. And we'll take some examples from the past papers as well as we move on, and you'll understand how exactly they have been asking those questions. All right. So that's why these are two types of questions that they ask in case of biotechnology. And in the mains examination, the questions have been fairly simple. Questions have been largely based on everything related to the various sectors and all the advantages, disadvantages, applications, etc. So these have been easy questions. So let's start with our chapter. Now, first, to start with, we need to understand what do we even mean by biotechnology. All right. So we need to break it down into two words. Just 1 minute, please. I hope my voice and video are clear. All right. Huh. So first and foremost, let's break down this word that is biotechnology and try to understand the meaning of this word. Now, you can say that there are two words here, one bio and second technology. Now, bio here, I mean, you have studied biology, so you know that bio is related to what? What is bio related to? What do you understand whenever you see this word bio? Yeah, life, living, correct. So overall, everything related to life, and that's why biology is the study of the living or study of life. So bio comes from a word which is bios. All right, it has its origin in the word bios, and this is where basically bios means something which is alive, something which is living. So it comes from here. So that's why biology, when it comes to it, is study of the living. Now, when we say biotechnology, okay, tell me what is the meaning of technology? We are studying the subject of science and technology, so I hope you understand what is technology. Can you, in one line, define for me what do you think? What, according to you, is technology? What is your idea of technology? Hmm. Development, innovation, something that makes life easy. Okay. The basic definition, Rohit, what you have given is the perfect way to define what is technology. Technology is nothing but application of science. All right. So when you say technology, the meaning of technology in simple terms is that it is nothing but the application of science. So we study physics, chemistry, and biology, and wherever you are applying physics, chemistry, and biology, that is what technology is. So space technology, we saw a lot of application of physics largely, correct? In case of energy, we saw a lot of application of chemistry. Now we come to biotechnology, where we'll see a lot of application of biology. So in this case, what happens is that all the basic sciences that you know, when you start applying that basic science in your real life for some kind of application or the other, then in that case you say that it is nothing but technology. All right. So that is what we mean by biotechnology. So now when you combine these two, what you can say is that here we are talking about application of the living organisms and their biological processes in order to make the best use of living organisms for the benefit of mankind. So this is what we define biotechnology as, that here we are talking about living organisms or their processes, living organisms or their processes in the benefit of the mankind. Now, what do we mean? What is the meaning of saying that it is for the benefit of the mankind, application of the living organism? A very simple example in our day-to-day lives with regards to biotechnology is making curd. Making curd from milk is an example. How exactly that it happens? You take a bowl of milk, you just add 1 spoon of curd in it, and just leave it overnight. What you'll see next morning is that that milk would have converted, if let's say the temperature is the summer months, milk would have completely converted into cold, into curd. In cold temperatures it might take a little longer time, but eventually what happens is that this milk will convert into curd. Now this conversion of milk into curd is what we uh, call as biotechnology, because here there is an application of a bacteria called as lactobacillus, which is present in the curd, and when it goes to milk, it finds its medium where it can start multiplying. It starts converting milk into curd. So that's why there is a living organism which you are using for an application, and hence making of curd from milk is an example of biotechnology. So that's why it is something that we have been using for a very long time, correct? As a technology, you can say that this is something that we've used for such a long time without even knowing that this was biotechnology, maybe for thousands of years. And apart from that, there are others also, other examples as well, like using preservatives, using a lot of natural substances as preservatives. It is also an example of biotechnology, because you're using something which is living or an application of it for preservation. Another example is the process of fermentation, which leads to the making of alcohols. And interestingly enough, if you go back into the history, you'll find that we understood how to milk alcohol before understanding how to make curd. So, in a manner, you can say that humankind has had its priorities straight. So, first came alcohol, and then later they understood that okay, curd can also be made from it. So, so that's why all these are examples. Even making of bread using yeast for making bread, all these are examples of biotechnology. Now, overall, what happens is that when it comes to biotechnology, these all are examples which have been examples where you can say it was hit and try, that somebody just stumbled upon something, that somebody, for some reason, milk converted into curd, and they realize, oh, this is also something which is edible and something that can be done. Similarly, the process of making bread, or even let's say alcohol, etc., all these things have been a practical approach, or maybe you can say a hit and trial approach, where they stumbled upon things like these. Now, overall, when we talk about biotechnology, these have been the things that have been in the past. But when we talk about the modern biotechnology, when we talk about the biotechnology of today, this is very different. This is not where we are just stumbling upon something. This is where our research, our studies, our understanding of innovations, all these are leading to newer developments in biotechnology. And this all specially started in 1950s. In 1950s, when for the first time we understood or discovered DNA, we understood, had a better understanding of the DNA and the genes. This is where we see that modern biotechnology starts to develop as a branch. Until now it was not really a big branch of technology, but 1950s onwards, as our understanding of the genes and the DNA starts becoming better, this is where we see that we start becoming better at understanding how to make the use of biotechnology in various different fields. And today we see that there are n number of sectors where biotechnology becomes useful. And this is precisely what we are going to discuss in this chapter, about what exactly do we mean by biotechnology and what are the various sectors of biotechnology. But before that, let's go into a basic understanding of where it all stems from. So as I told you, that it all started when we developed a better understanding of our genes and the DNA. Now, when it comes to genes and DNA and chromosomes, etc., also try to understand the meaning of what they are. All right. So first, let's try to see how they are and where they are located. Now, when you think of a living organism, an organism is made of organ systems. Like you might have heard of digestive system, skeleton system, circulatory system, nervous system, etc. So an organism is made of organ systems, and all these organ systems are made of organs. And there are various organs in our body. For example, stomach, stomach is an organ, heart is an organ, brain is an organ. There are a lot of organs in our body. Almost 78 to 80 organs are there in our body. So all these organs are set of tissues which perform a certain function. So these organs are made of tissues. These organs are made of tissues, and these tissues in turn are made of cells. These tissues are made of cells. So in this manner, we see that this is what the organization of a living organism is. Now, for the cells, what we say is that cells are the smallest structural and functional unit of life. All right. We say that cells are the smallest structural and functional unit of life. Smallest structural and functional unit of life. Now, why do we say so? Why do we say that a cell is a smallest structural and a functional unit of life? Now, the reason for us to say this is the fact that cell in itself is also, you can say in a manner, a complete organism. You might also know that there are various unicellular organisms, a lot of bacteria, virus, etc. They are unicellular. Unicellular meaning what? That they consist of only one cell. That's it. They just consist of one cell. And that one cell in itself is whole enough that it takes care of every functionality that organism needs for its survival, because it has various different cell organelles. Inside the cells there are various organelles, like there you might have heard of mitochondria. So do you know what is mitochondria? I'm sure that there is one term that is about to come, and many of you are about to give it in the chat. Yes, powerhouse of the cell, correct. So why do we say that it is powerhouse of the cell? Why do we say that mitochondria are the powerhouse of the cell? Yeah, ATP production, meaning energy, correct. So in a manner they provide energy for the entire cellular activity. Any activity that the cell has to have, for that the energy will be provided by mitochondria. So similarly, there are other organelles, and all these organelles together, they make a complete function out of this cell. And that's why we say that a cell is the smallest structural and functional unit of life. Now, inside the cell, there is a very important part which is called as the nucleus. Inside the cell, we have something called as the nucleus. And this nucleus, and what is the function of nucleus? We also say that nucleus is like the brain of the cell, right? We say that it's like the brain of the cell because it takes care of all the, it controls and coordinates all the activities in the human body. So, or let's say not only human body, in any organism. So that's why it is the brain of the cell. It controls and coordinates all the activities of the cell. All right. So in that cell, in that sense, that's why nucleus is called as the brain of the cell. Now, after this, when it comes to the nucleus, inside the nucleus, we have something called as the chromosomes. We have chromosomes inside the nucleus. These chromosomes, they are a set of bunch of different genes, and these genes are segments of DNA, deoxyribonucleic acid. So this is what we see inside the cell. Now, overall, this is basically the genetic organization. So overall, we see that this is how we look at inside the cell, and this dna becomes the smallest part of this entire nucleus, which contains all the information about an individual, about an organism. Every bit of information, every bit of character that this organism has is defined by what's there in the dna. And that's where this dna becomes very, very crucial, because in a manner you can say that these are like memory units. These are like memory units, just like how you have memory chips or storage units, like let's say memory cards or pen drives, etc., which can carry all, which can carry all the information. Similarly, you can say that, or let's say the basic storage that any computer has. If you think of any computer, laptop, there will be a storage that that computer will have, which stores all the information about that computer, plus everything externally that you keep filling in. So all those things will be there inside these memory units. So similarly, in the dna, all these characters of an individual will be defined by what's there in the dna, or what the set of dna is like. So that's why we see that this dna becomes the smallest part of this entire organism, which will define the different characteristics of that organism. And depending on what kind of dna are present, what sets of dna are present, the characteristics of those organisms or any organism will be defined in that sense. Now, these set of DNA, a bunch of DNA together, will make up a gene. All right, a set of DNA together will make up a gene, and that gene will have specific characteristics. It will define specific characteristics. So everything, I mean, for example, let's say color of the eye. There is a gene for defining the color of the eye, which is nothing but a set of DNA together, bunch of DNA coming together, making that gene, and this gene will be responsible for a specific color of the eye in a particular individual. So that's why every character will be defined by all these important parts inside the nucleus. So this is how we understand and look at the living world. If you go above this, I mean, these organisms, basically, there are populations of these organisms, and these populations, when they live together, they form communities, and these communities, when they come together, they form ecosystems. But this is a part that you are going to learn in your environment and ecology class. So in this manner, we see that this is how we are looking at the structure of the entire living world. uh, on one end, we are looking at the environmental structure, the ecosystem structure, then the living organism structure, and then the cellular structure. So overall, this is basically overall, this is what we look at when it comes to the entire thing. Now let's try to understand one by one that what is a DNA, what is a gene, what is a chromosome, etc. Now first, to start with, what do we mean by a chromosome? So in case of a chromosome, we say that these are long coiled up thread like structure made of DNA and proteins. So long thread like structures. Now in, for example, in human body, there are a total of 23 pairs of chromosomes. There are 23 pairs of chromosomes, and these 23 pairs of chromosomes define everything that a human is, or a human does, or what a human defines. So that's why all these things will be characterized by everything that we see in the chromosomes. So there are 23 pairs. Now, in humans, for example, out of these 23 pairs, meaning a total of 46, out of these 23 pairs, 22 pairs are common across the males and the females. Only the 23rd pair is where you see a difference. 23rd pair is where you see a difference, in the sense that 23rd pair defines whether that human will be a male or a female. So that's why all these 23 pairs, they define the entire character set of that human. So that's why these are long thread like structures. As you, if you go back, like, uh, it looks like this. So as you can see here, so these are chromosomes, all right? These are chromosomes which are found inside the nucleus of a cell. So as we discussed, that you have a cell, inside the cell you have a nucleus, and this, inside the nucleus you have these chromosomes, and inside the chromosomes you have all these DNA, and a segment of a bunch of DNAs together will make up the gene. So there will be a particular gene which is made of a certain number of DNA which is defining a certain character, and all these genes together, they will ultimately make up the chromosome. So we have the chromosomes, and chromosomes all carry all the genetic information in the form of genes and is found in the nucleus of a cell. Now, in terms of chromosomes, what we see is that when we break down chromosomes, when we look inside the chromosomes, we'll find that they are made of various different genes in the given body. So these are segments of DNA that contain the instructions for the development and function of living organisms. Now overall, what happens is that these genes, there will be a lot of variations also that can come in the genes, and all these variations are defined by the combinations of DNA inside them. So the combinations of DNA will define what kind of gene a certain organism has, and that will define the property, that will define the character. So a certain gene will be responsible for something. If there is any change that happens in that gene, then that character will also change. So that's why even when we look at diseases, many a times we try to look for certain genes, that if let's say there is some kind of problem that has arisen in a gene, or maybe some defect that has come up in the gene, and that defect is causing something, and these genes will have variations, and that's why uh, everything, for example, let's say the height, the color of the eye, the color of hair, all these things are defined by genes and the variations, which are also called as alleles. So in these, when it comes to genes, we discuss about chromosomes, that there are 23 pairs of chromosomes in the human body. When you look at the number of genes, there are almost more than 20,000 genes that we have found in the human body. So that's why all these genes will make up these chromosomes. And then lastly, we have the DNA. So DNA basically is a molecule that carries the genetic instruction used in the growth, development, functioning, and reproduction of all the living organisms. Now, when it comes to the DNA, as you can see here, it is like a twisted ladder, correct? If you take a ladder and you twist it, it is what the structure of a DNA is. And there are four chemical bases that we have. There are four chemical bases that we have. We have adenine, thymine, guanine, and cytosine. These are the four chemical bases. Now, adenine always combines with thymine, and guanine always combines with cytosine here. And these two will then combine. So they can form various combinations. So for example, ATGC can be a combination, TAGC can be a combination, TACG can be a combination, ATCG can be a combination. So this, like this, there will be various combinations, and all those combinations together will make up the genes. Now this entire thing, just think of a book. Like when you think of a chromosome, then chromosomes are like chapters in a book. That we are saying that there is a book which is the cell of, or rather the nucleus. It has 23 pairs of chromosomes, meaning that there are 23 chapters of this book. And when you divide these chapters, what you see is that there are various portions, there are various, let's say, sentences, or there are various parts of a particular chapter. So all those parts that you have in a particular chapter, all those parts basically become the genes. And then inside them, when you are looking at a certain word that you have in making those sentences, that word itself is a DNA. So that's where those words, when they are being defined as different words, what happens is that a word only has a meaning when all the letters come in a certain way. Correct? That, let's say, if you have three letters with you, A, B and C, it doesn't make sense as a word when A, B and C are together. But if you take C, A and B and make it cab, then it makes sense. Correct? It makes sense. And then in that case you can say that this is fine. So that's why DNA, when they come in a certain way, when they combine in a certain way, then only they start making sense, then only they add a property to the required organism. So that's why we see that DNA becomes the smallest. You can say that these are like letters, and the words that are coming out of it, the words, the meaningful words that you're making out of it, are the genes. And all those combined, they would make up a chapter. And there are 23 such chapters that are there in the body. So in this manner, that's where DNA is the smallest, then comes genes, and then comes the chromosomes. Now, apart from DNA, there is another such ribonucleic acid which is called as RNA. Now, RNA also has a very important function in the body, because everything that the body has, every protein that the body makes, all these are made of RNA. And hence, RNA become very, very crucial in the entire functioning of the human body, or not only human body, for any organism, because they will carry all the instructions required for making any part of the cell, or making any protein that is required for making different parts of the cell. So all these things will be done by the RNA. So that's why DNA and RNA, these two become the very basics. DNA will define all the characteristics. RNA will ensure that everything that the body needs, everything will be made by the RNA. And that's why they will be responsible for the development and the growth of all the things that are there inside the cell. So that's why these two become very crucial. And also, because they are chemically very similar, also understand some of the basic differences between them, especially from your prelims perspective. Something like this can be important, that what is the difference between DNA and RNA. Now understand the basic meaning first in the structure of DNA and RNA. Structure wise, what we are saying is that DNA, basically as a structure, is a double stranded molecule with a long chain of nucleotides, and RNA is a single stranded molecule with a shorter nucleotide. Now what we mean is that when you look at a DNA, what we discuss is that you take a ladder and you twist that ladder. So that twisted ladder structure is the structure of the DNA. Whereas when it comes to an RNA, it's like having half of that ladder. So that's why, as you can see here, that when it comes to the RNA, it has just one part of the ladder, and when it comes to the DNA, it has both the sides of the ladder. So that's the basic difference between DNA and RNA. So overall, structure wise, this is where the difference lies, that one is deoxyribose or nucleus, and second is it has ribose, and one has deoxyribose. So that's the basic difference in the structure. And then the difference in their function wise, in case of DNA, we know that all the genetic information, all the specific characters are there inside the DNA. Correct? All the genetic information is there in the DNA. And when it comes to protein production, which is required for the growth and development of a cell, all that will be there inside the RNA. So that's the basic difference in their function. And when it comes to their location wise, we see that DNA is found in the nucleus and mitochondria in the cell. And when it comes to RNA, it is found in every part of the cell. So every part of the cell where some kind of protein synthesis is happening, in all the parts of the cell, RNA will be formed. And not only nucleus and mitochondria, in case of DNA, it is also, when it comes to plants, in case of plants, it is also found in plastids. And even this has been asked in your UPSC prelims examination back in 2004. In 2004 prelims, there was a question where they had given these three: uh, nucleus, mitochondria, and plastids. And then they had asked which of the above have DNA in them. And then the options were one only, one, two, and three, two and three, etc. only. Now this is where a lot of people got it wrong, because a lot of people did not realize that it's not only the nucleus that has DNA, but outside the nucleus also there are two other organelles where DNA can be found, and one of them is mitochondria, other one is plastid. But there is a difference between the DNA that you find inside the nucleus versus the DNA that you find in mitochondria or plastids, in the sense that the DNA that you have inside the nucleus, that is specific to that organism, that is specific to that particular individual. But the ones that you find inside plastids or once you find inside mitochondria, they are not specific to an individual. All right, they're not specific to an individual, but rather they are actually common across the species. For example, the DNA that my nucleus has is different from the DNA that your nucleus has, but the DNA inside my mitochondria and your mitochondria are the same. If, I mean, considering that I am also human and you are also human, and no non-human entities are sitting in the classroom. So for all the humans, the mitochondrial DNA will be similar, and uh, for each one of us, the nuclear DNA will be different. So same goes for even the DNA found in the plastids, that they will be common across the species. So that is where the difference lies, in the kind of DNA that is present in the different parts of a cell. And then in case of the four bases also which are present, we discussed in DNA that you have adenine, guanine, thymine, and cytosine. Whereas in case of RNA, there is one difference, that you do have guanine, adenine, and cytosine, but instead of thymine you have uracil. So that's a basic difference between the chemical bases that are present in ribonucleic acid. So this is what you can say makes the base of everything, or all the characters. So that's why DNA, just understand that DNA, when it combines in a logical way, makes a gene, and gene will be responsible for anything, every specific characteristics in our body. For example, the color of the skin, the color of the eye, hair, or but the height of an individual, the shape of the face of an individual, how much of hair is left in on the scalp, all that will be defined by genes. So that's why if you have those genes where the hair has to go, then the hair will go. So all these things basically are defined genetically. So overall, that's why have a basic understanding of the differences that you have between the DNA, RNA, and so DNA, genes and chromosomes. That when we say that DNA is the basic building block, genes are segments of DNA, and the chromosomes are the ones that are making the complete, or which are storing the complete set of genes. So this is how we can understand the difference between these three, that is DNA, RNA, DNA, genes, and chromosomes. Before moving further, any question if you have, anything if you want to ask? What is the difference between organ and organism? organism is made of bunch of organs. For example, you are an organism, I am an organism, and we have many organs in our body. Heart is an organ inside the body, brain is an organ inside the body, stomach is an organ, liver is an organ, kidney is an organ. These are all organs inside the body, and all that makes organ systems, and then organism is a set of all these organisms or organs. Virus have RNA? Yes, virus do have RNA. There are two types of viruses. Either it can be a DNA based virus or an RNA based virus, and that too there can be single stranded DNA virus and double stranded DNA virus. Similarly, there are differences in the RNA viruses as well. So there are various different types of viruses. So yes, they do have RNA, but not all of them. They can either be DNA based or RNA based. What is genome? Genome basically is the set of all the chromosomes together. All the chromosomes together makes up the entire genome of an individual. So that entire book, you can say, is the genome. As we discussed that the chapters are the chromosomes, and all the sentences, or let's say all the words are genes, and all the letters are DNA. Just think of genome as the entire book. Please explain RNA once again. So in case of RNA, what we are saying is that RNA will be responsible for largely making proteins in the cell. So wherever protein production happens, now protein is required for growth and development of the cell. So wherever protein production is happening, you would need instructions from RNA. RNA will have all the instructions in them in how to construct or how to make these proteins in the various parts of the cell. What is mitochondria? So in case of mitochondria, just remember that uh, every energy or any energy that the body needs, all the energy will be required, I mean all the energy will be present inside the will be provided by the mitochondria. So mitochondria, that's why it is called as the powerhouse of the cell, because it will carry all the information that is required. It will have all the energy that various parts of the cell would need. Hmm. What are nucleotides? So nucleotides, basically all the chemical bases that we discussed: adenine, guanine, thymine, cytosine, uracil, all these are called as nucleotides. And so that's why they become the basic building blocks. These are nucleic acids, and they become the basic building blocks of the DNA. DNA in cytoplasm and ribosomes are same across Homo sapiens. See, DNA will not be found floating in cytoplasm. DNA will always be found inside the nucleus or inside mitochondria in animals, and inside plastids in the plants. Gene is a segment of DNA, so it should be the smallest unit. No, no, no. We are not seeing segments in that sense. We are saying that, see, there are a lot of DNA. Suppose that there are these many DNA, and out of these only these DNA are making up a gene. So a segment of the entire DNA set, that segment is making up the gene. So that is what we mean. So that's why no, gene is not smallest. What is the difference between chromosomes and mitochondria? They're completely different. You are, I mean, how do I even tell you the difference? There's no similarity in them at all. Does that mean we have three DNA? No, 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 no. We have millions of DNA. We have millions of DNA which are located in three different places. I mean, two different places for us. What is gram positive and gram negative? This is a way in which we define bacteria. We classify bacteria in that sense. Uh, I think it's better that we discuss this in diseases chapter. Corona had RNA. Yes, the virus has RNA. In simple words, what is DNA, RNA? In simple words, DNA has all the information about you, everything that you are. It's because of your DNA, and RNA makes up your body. RNA makes up the proteins for your body and hence constructs your body. Mitochondria and peptides are also inside the cell, not outside the nucleus. Yeah, inside the, but outside the nucleus. Yes, you are correct. Unicellular made of a single cell or more than one similar cell? No, unicellular. Uni means one. So unicellular means one cell. Is uracil connected to adenine by a hydrogen bond? Yes, it is. Please repeat again where in humans DNA is found. In humans, DNA is found at two places. Basically, uh, one basically found inside the nucleus, and the other one found inside the mitochondria. The difference in nucleotides and nucleosides. So the basic difference between nucleotides and nucleosides is that in case of nucleosides, you only have a sugar and a base, whereas in case of nucleotides, you have the sugar, you have the base, and at the same time, you also have the phosphate group which is attached to it. So that's the basic difference between a nucleotide and a nucleoside. In NCRTs, it is written chromosomes form only when cell division happens. Please explain. So basically, when we say that there are set of chromosomes, I mean there are pairs of chromosomes. So these pairs are found or formed whenever cellular division happens inside the body. So that's why every cell will keep on having a copy of this all these chromosomes with whenever cell division happens. Different organs of the body are made of cells. Does this imply all the cells of different organs of a person have the same 23 chromosome pairs? See, these 23 chromosome pairs will be present everywhere in the cell, I mean, every part of the body. So, every cell will have these pairs and they will have the similar pairs available everywhere. Is the new space policy 2023 important? Uh, wouldn't say so, because by the time the examination comes, a new space policy will be available for 2024. How to get access to new batch? Okay, I'm not getting option to change the batch under the switch batch option. This I'm not sure. It will have to be redirected towards the support team. Here, how will they ask questions? They can ask questions on various aspects. They can ask you questions based on the basic definitions, on the differences and the various aspects of, for example, between DNA and RNA, or between DNA and genes. So they can do that, or they can ask you to define the things. Difference between gene and genome. Genome, as I told you, that genome is the entire set of genes that you have inside, entire set of the chromosomes that you have form the genome. So genome is, you can say, which contains everything. The entire set of chromosomes together will become the genome. Which NCRT books biotechnology chapter available? It's there in class 11th and 12th, but you should not read that. It's very technical in nature, so not required. There are so many questions. But many of these are now repeated questions. Changes can be made in plants. Likewise, can we change characters and skills of humans by making changes in DNA? This is what this entire chapter is about. We'll discuss that later. Sugar, base, and phosphate in present in nucleotide or nucleoside? Nucleotide. Nucleoside does not have phosphate. Nucleotide has the phosphate. Do they ask direct questions? Yes, yes, they do. It's not a written rule that they will always ask indirect questions. Many times we actually, you have raised a good point. Many times we see this, that, for example, while making questions, many a times we also make simple questions. Then there will be one student who always comes and says that, why are you asking simple questions? They will never ask you a one line question. Just go back and look at the question papers. There are one liners also being asked. There are complex questions also being asked. They are using various different patterns to ask the question. So it is not defined that they'll always ask you three statements, and then they'll ask you to pick which are the following are correct, or how many are correct. There are direct questions also. There are direct fact based questions also. So it's not that uh, it's not, you should not judge what kind of questions can come or cannot come. You have to be prepared for everything. All right, so let's move on. Now we come to what this chapter is really about, and this is about all the different branches of biotechnology. Now, until now, we have just understood the basics of biotechnology and of what the body is made of, what an individual is made of, and everything. Now, here onwards, these terms that we just saw, these terms is something that will keep on using in all the in this entire chapter. Now, what we are going to study in this chapter is about applications of biotechnology in various different sectors, and how we can actually make the use of biotechnology. So first, when we look at the various branches, there is a way in which we do a color coding of the branches. Like we have green biotechnology for agricultural applications, red biotechnology concerned with medical science, blue biotechnology concerned with water related things. So anything underwater, anything related to water will be called as blue biotechnology. Similarly, we have white biotechnology for industries, gray biotechnology for the environment, brown biotechnology for anything that is applied to the arid zones or the deserts. And then we have violet biotechnology, which is related to anything outside the sector. So, for example, the laws related to biotechnology, the acts related to biotechnology, the intellectual property rights, the laws, the contracts, each and everything related to, I mean, not a core branch, but everything else. The documents and the law, the ethical issues, the moral issues, the philosophical aspect of biotechnology, all that will come under violet biotechnology, and then there is something called as dark biotechnology, which is more about the misuse of biotechnology. This is about the misuse of biotechnology. So in this sense, there are a lot of ways in which we can look at the various branches of biotechnology, and this is precisely what we are going to study in this particular chapter. So we'll start with all these branches one by one, and we'll go into the details of each and every branch and try to understand the various aspects of that particular branch. So the first branch that we have is medical or red biotechnology. Now, when it comes to medical applications of biotechnology, this is where you can say all the money in biotechnology goes. All the investments in biotechnology goes in medical applications. So that's why you'll see that a lot of money. For example, let's say in case of India, if you look at the data for India, you'll find that 70% of the money spent on biotechnology in India is being spent just on one branch of medical biotechnology, that is uh, related to drug administration, or or something that is related to biopharma. So in case of biopharma or biopharmaceutics, you'll find that almost 70%, just imagine that out of 100 rupees being spent on biotechnology in India, 70 rupees is only being spent on biopharma. So that's why that tells you the importance of medical applications of biotechnology. And it's not only a figure for India. You look at the worldwide figures, the budget allocations, and every money being spent, or any every penny being spent on biotechnology, you'll find that majority of that money flows in medical applications, because we know that with the help of biotechnology we can solve the unsolvable. We'll be able to solve the issues that we haven't been able to solve until now. We'll be able to find cure for diseases that we did not know existed. We would be able to make better vaccines, more effective vaccines, because of biotechnology. So that's why biotechnology in the medical science specifically becomes very, very crucial. And that's the reason why so much of money, or why a large portion of the money spent on biotechnology is spent on medical applications. So the first medical application is gene therapy. Now, in case of gene therapy, we are talking about the uh, use of genes in curing of a disease in one way or the other, to use the genes to uh, cure a disease. Now, how exactly can we do that? Many a times, what we'll see is that when it comes to gene therapy, gene therapy becomes specially useful when you are looking at a disease for which we haven't been able to find a medicine, we haven't been able to find a vaccine. So in all these diseases, we have been trying to find the ways in which genes can be used. Now, how can we make the use of genes? What we discussed is that genes are responsible for certain characters, that every individual character that we have, it's because of a certain segment of DNA that would have made a gene. Now, sometimes there can be a defect in the gene. All right. Sometimes there can be a defect in the gene because the DNA may not have combined properly. So there is a defect in the gene. Now, because of that defect, the work, the function that that gene had, it may not have been able to perform it well. So it may happen because of either absence of a gene altogether, that a gene which was responsible for something certain is altogether absent. So that's where that characteristic is itself absent, and that may have been causing a disease. So either it could be because of absence of a gene, or maybe presence of a gene, presence of an unwanted gene. So either there is an absence of a required gene, or there is a presence of an unwanted gene, a gene that is causing the issue because of its presence. It should not have been present. Or maybe it also happens because of changes that happen in the gene. So these all these can lead to diseases. All these can lead to diseases, or they can form various problems. So overall, that's why this is what happens. Now, what do we do when, let's say, we see that there is an absence of a certain gene? What do? We add a gene. The solution to this will be to add a gene. That will have to look for a certain gene like this, and we'll try to add that gene to the structure of that organism, or in this case humans. So adding a gene which was required can solve this problem. Then in this case it could be about removal of a gene. So we can remove the gene which is causing the problem. If, let's say, it has no positive impact on the body, then probably we can even remove it, and that removal itself will help us in solving that problem. And changes in a gene. So changes, whenever changes happen in a gene, then in that case we can modify the gene. So you can say that in a manner there are these three mechanisms that help in case of any disease that where we do not have a medicine or we do not have a vaccine, etc. In all these cases, these are the ways in which gene therapy, any one of these processes being used, addition of a gene, removal of a gene, making modifications in a gene, replacement of a gene, all these things can be called as gene therapy. All right, all these will be called as gene therapy. So I'm just writing it here. So in a manner, gene therapy includes includes addition, removal, replacement, or modification of a gene. All right, so this is what gene therapy does, and this is what will help us in preventing a disease. So to prevent or cure a disease. So this is what we do with gene therapy. Now, apart from gene therapy, we have another important aspect which is called as genetic testing, where we test for a certain gene to find a problem. So we look for certain genes many a times and see if that gene is present and is intact. Is there any modification that has happened in the gene? Is there any change that has happened in the gene? Is that gene present at all or not? So all these things also are a good indicator of a disease, especially, let's say, when it comes to a genetic disease. Many of these diseases, which are genetic in nature, which have been caused because of some defect in the genes, these are also passed on from generations to generations. That if the parents have that genetic disease, that may be passed on to the child as well. So that's why location detection of that genetic disease is very, very important to solve that problem. So that's why genetic testing becomes useful here. At the same time, many a times you will see that even early detection of cancer is possible, or a chance of a onset of diseases like uh, diabetes is also possible. So in all these cases, what happens is that they'll test for certain genes, they'll try to find certain genes, and those genes will be analyzed. And if those genes, after analysis, they do not show any problem, then probably the person is safe. But if there is some problem that that analysis shows, then maybe that person needs to take some kind of help. So that's why testing of the genes also becomes very, very crucial. That even today, for example, let's say, there are so many full body checkups, etc., that are now available. There are so many companies which are doing regular lab tests, and they can come to your home, they collect blood samples and go. So there you'll see that they can do the test for even finding out whether chances of diabetes is there, or finding out chances if the cancer onset is present. So all present, all these things can actually be found. And in all these cases they'll be looking for certain genes, and they'll be looking for certain genes and analyzing those genes to find a present or a future possibility of an onset. So that's why the results of a genetic test can confirm or rule out a suspected genetic condition, or help determine a person's chance of developing or passing on a genetic disorder. So in all these cases we see that genetic testing becomes very crucial. Then in drug administration, now in drug administration we have a branch which is called as biopharmaceutics. Now this biopharmaceutics as a branch becomes very very crucial, because this is where the money flows. This is where we have seen that the huge amount of money investments are happening in biopharmaceutics, because this is where we are trying to find solutions or find cures or medicines against diseases where we are struggling, where we haven't been able to find something, we haven't been able to find a cure. And even if we have, we are still trying to find better medicines, faster medicines, cheaper medicines. In all these cases biopharmaceutics becomes very very crucial. So that's why, so when it comes to making of the pharmaceutical drugs and make the use of biotechnology in making drugs, this is called as biopharmaceutics. Pharmaceutics, you might know, is related to medicines. So biopharmaceutics, when biotechnology is being used to make medicines, so that's why biopharmaceutics. So that's why it examines the interrelationships of the physical and the chemical properties of the drug, the dosage form, and the manner of drug administration on the range and extent of drug absorption. So that's why biopharmaceutics is where we are making the use of biotechnology in making effective medicines or in making effective drugs. So then it is called as biopharmaceutics. Now, in case of biopharmaceutics also, there are various aspects of biopharmaceutics. For example, there is something called as pharmacokinetics. All right, there's something called as pharmacokinetics. Now, if you break down this word pharmacokinetics, first word is pharma, right? The first word is pharma, and the second word is kinetics. So, first word is pharma. Pharma is related to what? Medicines. Kinetics is related to what? What is kinetics related to? Yes, movement or motion. So there are two words, medicine and motion. So in case of pharmacokinetics, we are looking at when you take a medicine, how long does it take for your body to start absorbing that medicine, and then the medicine to start taking effect on your body, and how long does it take for this medicine to entirely be excreted out of the body. So when we are looking or studying about all these rates, the rate of absorption, the rate of excretion, the rate of and the rate of how fast the medicine starts working. So in all these cases, when we are looking at the motion, when we are looking at the speed, or the movement of the medicine inside the body, this is where we say that we are talking about pharmacokinetics. So that's why pharmacokinetics becomes very crucial, because every disease has a different way in which it onsets and a different speed at which it onsets. So for example, let's say there are diseases like cholera or diarrhea, etc., where the onset is very fast. It could be within hours, within couple of hours of time they can onset. Whereas there are other diseases which can take a lot of time. Let's say leprosy as an example can take up to 5 years of time to develop. So that's why every disease has a different speed of its onset, which is called as incubation period. So incubation time periods are different. In case of COVID-19, for example, we knew that it could start taking effect in 4 to 5 hours, and it could take up to 14 days of time to start developing the symptoms. So that's why the quarantine period used to be of 14 days, because if somebody has not developed the disease in 14 days, then the chances are that the person is safe. So that's why the quarantine period used to be of 14 days. So that's why it could take hours, some hours, to up to 14 days. So every disease has a different speed, and your medicine has to be faster than that disease. So if let's say the disease takes up to 2 days of time, your medicine should start taking effect within a day. If the disease takes 2 hours of time, your medicine should be effective within an hour. So that's why pharmacokinetics becomes very, very crucial in understanding the speed of absorption of the medicine and how fast it starts taking effect in the body. Then we also have something called as pharmacogenomics. Now, in case of pharmacogenomics, we are talking about a combination of genetics and pharmaceuticals. So, overall, how exactly a genetic makeup affects an individual's response to drugs is what we study here. That a certain set of genes, and let's say when you talk about the variations in the genes, we know that every part of the world there are some genetic variations. Like the genes in Indians in general will be different from the genes in a European or an Australian or an African, correct? So we know that there are certain differences that we have in the genetics. Now, how exactly these genetics if have an effect on an individual's response on drugs to individual's response to drugs is what we study as a part of pharmacogenomics. And that's why this also becomes very crucial, because how the various genetic variations can be taken into account while making a medicine becomes very, very crucial. So that's why uh, the gene expression with efficacy or toxicity of the drug is what we study as a part of pharmacogenomics. Then after this, there is something called as virotherapy. Now, viro here comes from virus, meaning when we are making the use of virus for any kind of medical application, we can say that it is virotherapy. Now, there are antivirals, for example, antiviral medicines which are actually made of virus, or in case of vaccines. In case of vaccines especially, you might know that in case of vaccines we do use virus, correct? We do use virus in making various vaccines. Even in case of COVID-19 vaccines, we know that certain virus have been used in making of that vaccine. So that's why that all will be called as virotherapy. When we are making the use of virus as a therapeutic agent, then in that case we'll call it as virotherapy. And then there is insulin. Now, insulin, you might know that insulin is present in our body and it removes excess sugar from the body, correct? It removes excess sugar from the body, and to remove that excess sugar, insulin is required. Now, what happens in case of diabetes? In diabetic patients, that insulin production is neither either not happening at all, or it's happening very slow, correct? It's either happening very slow or it's not happening at all. So then in that case, these insulin have to be taken externally. So that's where diabetic patients might take insulin injections, so that they inject themselves with insulin and it will help them in removal of excess sugar in the body. So that's why, because this is a part of the biochemical processes of the body, when externally or artificially insulin is being made, that also becomes biotechnology. So overall that's why we see that there are various different applications in which we can make the use of biotechnology in case of medical, or in case of medical science. Now, when it comes to let's say your examination, especially in case of your mains examination, what happens is that in the mains examination a direct question can come from here, that they can directly ask you what are the medical applications of biotechnology, or what is red biotechnology, what are the various aspects of red biotechnology. So in all these cases, what we see is that uh, question, either they can ask you on specifics, that what is biopharmaceutics or what is pharmacokinetics, and all these kind of terms can be asked in your prelims, or in the mains examination a direct question can come, that what is the health application, what are the various ways we can use it, what is the future, and all these things. And there are things that we have not studied that we are going to study later. For example, you have the stem cell therapy, which is a very important application in medical science. So we'll discuss later what do we mean by stem cell therapy. Then there is even the use of cloning. So how exactly we can make the use of cloning also in case of medical applications. So there are ways in which we can use it. Similarly, the use of DNA fingerprint. The use of DNA fingerprinting also can be seen here. So there are all these things, and that's why they can all be counted as a case of medical applications. The next is agricultural applications. Before that, if you have any doubts from everything that we discussed in medical applications. Pharmacogenomics not clear. So pharmacogenomics basically is when we are making the use of genes, or when we are taking the use of, or let's say, our understanding of genes to make a medicine. That, as I told you, that everybody has different genes, right? So when there are genetic variations, different genes might respond to medicines differently. So that's why when we make a medicine, we make sure that all the genetic variations are being accounted for, that a certain person's gene may not affect um, the use of that drug or the efficacy of that medicine. Many times you might come across these cases that there are certain medicines which are not used in certain parts of the world because they are useful for you, but the same medicine, let's say, being used on an African or used on an European, probably will not have the same effect, or it can actually have a side effect, and it is not giving you the result. So that's why different genetic makeups, they respond differently. So when we take into account the genetic makeup and make medicines accordingly, it is called as genomics. Stem cell therapy for anemia, yes, but we'll discuss that later. Cloning means, we'll discuss later. Cloning is a topic that we'll discuss properly. How many genomes are there in a cell? A cell will have, I mean, inside the nucleus there will be one genome. Is this the first class for new batch? No, no, no. The first class for the new batch, that is 2025 batch, is polity, and that is a different session. Can I say that mitochondria, which is also a cell, has DNA, but unlike chromosome, which has DNA nucleus, mitochondria has DNA outside nucleus? Yes, of course, because mitochondria does not have the nucleus in it. So yes, that's true. In thyroid, yes, gene therapy is usable there. Recombinant insulin not clear. So basically, insulin is something that our body makes, but in diabetic patients, insulin is not made by their body. So that's why we inject insulin. We make insulin outside the body and we inject insulin, and that's why it is also called as an application of gene therapy. Sorry, application of medical biotechnology. How pharmacogenomics different from biopharmaceutics? Pharmacogenomics, you can say in a manner, is a sub part of biopharmaceutics. So pharmacogenomics is only about genes, is only about looking at the genetic makeup of an individual and how the drug response in that individual is. But whereas when it comes to biopharmaceutics, it talks about everything, every kind of study related to living organisms and medicines. So that's why it is not restricted to only genetic studies. What is CRISPR technology? We'll discuss that later. We'll discuss it. You'll understand it later. So, in virotherapy, virus converts to medicine to kill another virus. Yes, that's true. What is the meaning of the term recombinant? We'll discuss that. Just wait, please. Who is taking polity? Anubhav sir has been taking the polity class. Why we need insulin? Insulin is required to remove excess sugar buildup from the body. Would you talk about genetic engineering and its tools? Yes, I mean, this is what the chapter is. We'll discuss that. How can we cure genetic diseases as chromosomes are present in every cell, so we cannot change or remove genes? So, see, uh, it's not about, let's say, only it's not about the entire set. All right, it's not about the entire body, entire set. Gene therapy will be used for certain particular. So for example, let's say gene therapy being used for a heart disease, so then we are only looking at the genes which are present in the heart or the muscles or the cells that are there near the heart, and that is what you're trying to solve. That's the only thing that you're trying to solve. So that's why you are not really bothered about the genes which are present in other parts of the body, only in the heart, and that is what you're trying to solve. This chapter important for prelims or mains? It is important for both. It is important for both your prelims and mains examination. All right. Can we cure hair fall by changing in genes? I wish there was something that could be done, but there are many novel methods that people have been trying to use. This became personal. I'm suffering from a disease called as Stargate. Okay. Leads to progress of, destruction, and doctor said can be cured by gene therapy. Okay, all right. Hmm. Indian Express had an article mentioning finding alternatives to bone marrow transplant and finding drugs to cure sickle cell anemia. Is that also application of biotechnology? Yes, it is. It is related to biotechnology. Hmm. So let's move to our next topic. This is agricultural applications of biotechnology. Now, in case of agricultural applications, this is one of the most important examples or one of the success stories of agricultural applications of biotechnology. Do you know what is this? Have you heard of this? I'm sure many of you know. Yes, it is cotton, and specifically, this is BT cotton. Now, BT cotton has been one of the success stories in agricultural applications of biotechnology. In case of cotton, almost 20-25 years back, the situation was worsening, where a pest called as bollworm, was affecting a lot of cotton crops. And it used to just eat up the cotton, the cotton flowers, and then it would completely destroy the production and drastically reduce the production. And for farmers all across the country, it was becoming a nightmare. And this is when we see that BT cotton gets introduced, a genetic modification in cotton gets introduced, where because of this modification, the cotton plant starts making its own pesticide, and it starts becoming poisonous for bollworm, and the bollworm is not able to eat upon cotton anymore. And hence we saw that there was a drastic jump in productivity. And that's why we say that this is one of the success stories of uh, biotechnology in agriculture. Now, overall, when it comes to agricultural applications, we are talking about something which is a transgenic plant. Now, when we say transgenic plant, transgenic, two words, transgenic, meaning that there is something called as a transgene. All right, there is a transgene. Now, when we say that there is a transgene, when we say that there is a transgene, understand the meaning first. Now, when we use the word trans, let's say when you say that somebody has been transferred, means what? That the person has now gone from one place to another, right? So that is what trans is. That when you have something from somewhere else, then the word trans there will be used. So in transport, because you are enabling movement from one place to another, to transport. So that's where the trans will be used, the trans word will be used wherever you are talking about something going somewhere. So when we say transgene, it means that you have a gene that has come from somewhere else. Now this somewhere else is another organism. So that's why this transgene is a gene from a different organism. All right, so transgene is a gene coming from a different organism. So, and it can be any organism. Suppose that for a plant, when we say that it is a transgenic plant, then it means that this plant has a gene which has been taken from some other organism. It may not be a plant, it could be a plant, animal, human, a microorganism, an alien, anyone. All right, any organism where you can take the gene from, you will take the gene from there, and you will insert that gene inside the plant to make, or let's say introduce certain properties, or maybe introduce certain better properties, or maybe take care of a disease or do something like that. So whenever we insert a gene from outside, whenever we take a gene from some other organism, then in that case we say that it is transgenic plant. So that's why there can be various different types of transgenic plants. BT cotton, for example, is an example of transgenic plant, because here we have taken a gene from a soil bacteria. We have taken a gene from a bacteria, and this bacteria had properties. This bacteria had genes which would make it poisonous for pests. So that's why we took this gene from a bacteria, and this gene was inserted inside the plant, the cotton plant. And then the cotton plant also responded, and it also started making a lot of these genes. Specifically, the name of the gene was CRY1AC. So that particular gene was taken from the bacteria, and from the bacteria, when we took it, we basically inserted it inside the plant, and hence we saw that BT cotton hence was made. So that's why you can take from wherever you want. I mean, you can take this gene from wherever you want, depending on what property you want to introduce to a particular plant. So for example, let's say you want your plants to walk. So then in that case, you will have to pick certain genes. You will have to see what are the genes inside the human body which are responsible for you walking. So then you have to pick those genes and then just insert those genes inside the plant, and the plant will start walking and start saying, I am Groot. But all these things actually do not happen. I mean, the things are not that simple that you can simply take a gene from anywhere and do anything that you want. Not everything works. So we'll have to first find what works and what does not. So for that, we need to understand that there is a certain way in which we can do things. So we'll discuss that. So there are various different desirable properties that you have for which you will produce a transgenic plant, for which you will introduce a genetic change in a particular crop. So for example, for increased crop production, and this is what we saw in case of bt cotton, that we have been able to produce or we have been able to increase the productivity, increase the yield of cotton by introducing this property. Then improving the nutritional value. Now this is also something that can be done, and an example here is of golden rice. If you look at the variations of rice, this is the normal rice, white in color, and the other one that you have here, which is yellowish in color, is the golden rice. We don't call it as yellow rice because yellow rice does not sound good. Golden rice sounds exquisite, so that's why we named it as golden rice. Now this golden rice basically has a property that we have enhanced the levels of vitamin A in golden rice. All right, so in case of golden rice would have better amount of, or rather it would have vitamin A, because in general rice does not have vitamins, right? Rice does not have vitamin A. What does rice have? Rice has carbohydrates. Rice has carbohydrates in huge quantity, but not vitamins, not vitamin A. But what we have done is that we have taken a certain gene from daffodil plant and inserted inside rice, and golden rice hence was made. And this particular gene that we have taken from the daffodil plant, it has something called as beta carotene, and this beta carotene converts into vitamin A. And hence, once this gene has been introduced to rice, rice also can synthesize vitamin A. So that's why consumption of golden rice would mean that you are also getting vitamin A apart from carbohydrates. So that is the difference that comes in here. So here also we found this gene where? In daffodil plant. We did not take it from another fruit or vegetable which is a good source, rich source of vitamin A. We took it from a flower, we took it from daffodil, and from there we took it and we inserted in rice and it worked. So then in that case, we can actually increase the nutrition value also. So golden rice, that's why becomes an example where we took beta carotene from daffodil plant, and this beta carotene is converted into vitamin A. The ones which are natural sources of vitamin A, the fruits and vegetables, they also have something called as alpha carotene, and this alpha carotene converts into vitamin A, and in this case beta carotene converts into vitamin A. So this is how we see that golden rice becomes an example. Then it can also be used to increase the shelf life of any given plant or any fruit, vegetable, seed, etc. For all of them, shelf life can be improved with the help of biotechnology. And then there are environmental benefits. For example, let's say if you even consider bt cotton, then in case of bt cotton, what we have done is that we have been able to also help environmentally. How? Because if bt cotton is making its own pesticide, then it means that you do not have to introduce chemical pesticides externally. So that's why the reduction in pesticide amount would also mean that there is a betterment of the environment, because pesticides are harmful chemicals. So they would then mix with the soil, mix with water, and contaminate everything. But then here in this case, you do not really have to use that much of pesticides in cotton. So that's why we see that there are environmental benefits also that they bring in. Now, overall, when it comes to the development of a transgenic plant, as I told you, that now in case of agricultural applications, as I told you, that these transgenic plants is where you have inserted the gene from some another organism. Now, does it mean that it is very easy to do? I mean, just can we just take a gene from somewhere, just insert it inside the plant, and it would all start working? Unfortunately, the things are not that simple. That there is a certain process that we have to follow to see whether or not it works. And this is where we have a technology called as the recombinant DNA technology. So recombinant DNA technology is where we are using the genes from another organism and inserting it inside the plant. So that is called as recombinant DNA technology. Now let's try to understand what do we mean by recombinant DNA technology. What happens in case of recombinant DNA technology? First, we isolate the required gene, the gene of interest. So this, for example, is the gene of interest for us. This particular gene, the blue colored part. This is the gene of interest. So first we have to locate this gene of interest. For example, this is the gene that was there in the soil bacteria for bt cotton, or the beta carotene containing gene which was there in daffodil plant. So that's the gene of interest for us. So first we isolate that gene of interest, we clean it up from any impurities or macromolecules that can be present on this gene. So first you have this gene in its pure form, and then this gene will be inserted inside a host. This gene will be inserted inside a host or a vector, or not host, let's say call it as a vector or a carrier. So this gene will be inserted inside this vector or a carrier, which can either be a virus or even a bacteria. Largely it will be virus, right? Largely we'll be using virus, but even bacteria can be used as a carrier or a vector. So now what we do is that we take this vector, and inside the vector this gene of interest will now be inserted. So now this insertion is also not that easy. It's not that you just do something, insert it. No, you will have to find where can you insert it. It's like a jigsaw puzzle. All right, it's like a jigsaw puzzle where you have missing pieces, and you'll have to find where exactly can you fit that piece. So unless you know where exactly to fit that piece, you can't do anything. Correct? So that's, it's not that you can just fit it anywhere you want. No. Correct? Similarly here also you'll have to find exact location where insertion is even possible. So then first of all you will have to choose that vector. Then you first have to look for that vector where the insertion is even possible, and then find the location in that vector. How exactly is it possible? And this is where genetic engineering tools come into the picture. Tools like CRISPR, if you have heard of. So tools like CRISPR, Cas9, etc. will help us in locating where exactly can we make the cut, where exactly can we find a suitable location. So suppose that a suitable location is found. So let's say this is where we found the suitable location and we made the cut. We made the cut in a manner that now you will cut it open. All right. So this you will open, and the gene of interest will be inserted inside it. So it's like this. That, let's say this is the part where you have made the cut, right? This is the part where you've made the cut. So this kind of a cut has been now made, and once you've made the cut in this particular gene, this is where, so this is the entire gene. Right, this is the entire gene. Now this gene of interest that you have right here, this gene of interest will be inserted here. All right, this gene of interest is now inserted here. So this gene of interest, so this one now is inserted here. Now this has been inserted, so now it has become a part of this genome. It has become the part of the genome of this vector. Now the next thing for us will be to make these ends to stick together. Correct? So this is where we use something called as DNA ligase. This is called as DNA ligase. So with the help of DNA ligase, we have uh, joined the two segments of the DNA, because on one hand you have the gene of interest. This is the gene of interest, and then here you have, this is the vector genome. So inside the vector, now gene of interest has been located, or it has actually been inserted. So for first thing that we have done is that we located a point where this cut could be made. So there also we use something which is called as a restriction enzyme. So you make the use of restriction enzymes, and these restriction enzymes will help us in understanding of this particular DNA, understanding of where to make the cut. So first, with the help of restriction enzymes, we will make the required cut at the required location. We cut it open. Gene of interest is inserted here. DNA ligase. Now DNA ligase is like, you can say, a glue. It's like your Fevistick or a Fevicol. So you are applying Fevicol here to make them the ends to stick together. So this kind of a DNA where you have a combination of these two DNA structures, the gene of the virus and the gene of interest. So that combined gene is called as the recombinant DNA or the recombinant gene. So that's why the term recombinant, because it's a combination of two different gene sets. So that's why recombinant. So recombinant, because this is where you have a set of two different genes, and once you have this, we create number of copies of it. We create thousands of copies or maybe lakhs of copies at times of this recombinant DNA. And once we have done that, this process is also called as PCR, polymerase chain reaction. So once we have created multiple copies, this is when we finally insert it inside the host or inside the required plant. So this is the entire process. It was not a simple process of you just taking a gene from somewhere and just inserting it inside the plant. This is the entire process that we follow, and this process is called as recombinant DNA technology. So what we are doing is, just to recap, what we are doing here is that we are first taking the isolating the required DNA in its pure form, and then after that we are cutting the genes in the recognition sites. So we take the help of restriction enzymes in determining the location at which the desired DNA is supposed to be inserted, and then the ligation will be done, which is joining of the two pieces, and this will be done with the help of the enzyme which is called as DNA ligase. So first isolation, then insertion, uh, first I mean location, and then insertion, and then ligation, and then after that amplification with the help of something called as polymerase chain reaction, where we are talking about thousands to millions of copies of the DNA being created. So we do that, and then after that, this recombinant DNA will be inserted inside the host, and this process is called as transformation. This process will be called as transformation. So in this manner, this is the entire process. This is the entire process that we use to uh, make the use of recombinant DNA technology in terms of making the changes in the plants in terms of producing the transgenic crops. Look at this question. This was asked in your paper. I'm launching the poll for you. Please mark your answers. Hmm. So 48% of you have marked D, which is actually the correct answer. Recombinant DNA technology allows the genes to be transferred. What are we making out of recombinant DNA technology? We are making the transgenic crops. And in transgenic crops, you have transgenes. Transgenes can come from anywhere, any organism, is what we discussed, across different species of plants, from animals to plants, from microorganisms to higher organisms. You can do anything. It allows the genes to be transferred from anywhere. There is no restriction. What did I tell you? You can take it from any organism as long as it is suitable. You can take it from any organism. There is nothing that is restricting you from not using it from any other organism. That is what the definition is. Transgene, this is what we discussed, that you take the gene from anywhere, any organism, microorganisms, plants, animals, humans, aliens, wherever you can find that gene. So that's why it allows the genes to be transferred from anywhere. So across different species of plants, yes, of course, but at the same time, even from animals to plants, it is allowed. It may not work. In a lot of cases, it may not work, but it is allowed. So yes, it can be done. And microorganisms to higher organisms, we anyway discussed that there is bt cotton. We just discussed about bt cotton. So I I mean, A and B should not have been marked, because C, I mean, statement number three is very crucial. And this is what we have been discussing all this while, that we took a bacterial DNA. Here also we saw that we use the bacteria. So you can use the bacteria, you can use the microorganisms. So that's why one and three for sure are correct. There can be a confusion in two, I understand that. But one and three, there shouldn't be any confusion at all. So correct answer will be D, that is 1, 2, and 3. Let's take a break here. If you have any questions, please post your questions. Let's take a break, come back, and I'll look at all your questions first, and then we'll keep moving on with agricultural applications of biotechnology. This is 7:45, so let's be back by 8 and let's continue. Welcome back, everyone. First and foremost, if you have any questions, let me start from there and then we'll move forward. Is this grafting of plants to promote vegetative propagation by using biotechnology? Not really. That's a very different process when it comes to vegetative propagation, because you do not know what will work, what will not work there. And in case of biotechnology, it's perfectly based on studies of what can work, and it's a completely a method which is genetic in nature, where you are making the changes in the genes at a microscopic level. Whereas grafting is a technique where you are trying to uh, look into the compatibility of two plants with one another. That's very different. What is the difference between genetic engineering and gene therapy? Genetic engineering is every time that you are making the use of genes for anything, any process. And gene therapy largely is a therapeutic process where we are talking about specifically medical applications. Whereas genetic engineering is a broader term. It is used in agriculture, in medicine, in all these different aspects. So it's not only related to therapeutic use of genes. When you make the use of genes in medicines, this is where you're talking about gene therapy. That's the basic difference. So in a manner, you can say that gene therapy is a sub part of genetic engineering. What is vector or bacteria in the first place? It's just a carrier. A vector, I mean a virus or a bacteria that can act as a carrier of that gene that is required. From last 2 decade onwards, there is increasing use of eating the chicken which has been grown by using medicines. Now we are facing some problems like our body made drug resistant for some bacteria. Likewise, is there any disadvantage of developing the golden rice or BT cotton 5 or 10 years down the line? Shouldn't be, because these are two very different cases. In case of let's say poultry, where you are talking about, there the problem has been the overuse of antibiotics. Overuse of antibiotics has caused it. Whereas in case of uh, genetic engineering in BT cotton or the other cases like golden rice, etc., we are not really using a lot of antibiotics, etc. So it should not have a negative impact as overdose of antibiotics do. So that's why these two should not be seen in the same light. Explain cryogene. I'm not sure what it is. Transgenic crops are banned in India. In food crops, yes. In food crops, we haven't really allowed it, but BT cotton has been used. So a transgenic crop has been used in India. But in edible crops, we have seen that it has to go through a lot of processing, and then only it is allowed. Then it is only it is put forward. Otherwise, not. So more or less, you can say it is banned. Only in a couple of exceptions, it hasn't been. What does microorganisms to higher organisms mean? Smaller to bigger? Yes, that is what it means. Basically, meaning from bacteria, virus, etc., to higher plants and animals. Does all DNA make up chromosomes, or some remain idle as well? Inside the nucleus, they will always make up the chromosomes. Although in microorganisms, that's not the case. In microorganisms, there is no chromosome. So the DNA, in case of the genetic material, they will just keep lying around that in that nucleus. BT mustard, or rather GM mustard, will uh, read a little later. Are we using DMH11? We'll discuss about DMH11 later. Just wait for some time. There was a bollworm pest attack on BT cotton recently over North India. Is it due to pest resistance of bollworm? Yes, it is. And there is a very good article published on it in Down to Earth. You just go on Google, type Down to Earth BT cotton Punjab issues. You'll find that article. Wonderfully explained everything that has been happening. So over a period of time, you know that every organism evolves, and this is something that we have started seeing in case of bollworm as well. That bollworm, despite you having BT cotton, it has now started affecting BT cotton also, and it is now affecting the entire productivity of cotton in especially the North Indian states where cotton is being grown, or BT cotton rather is being grown. So for details on this, I would say that just go, and there is an article on Down to Earth. You'll find it on Google. It's a wonderful article to read. How are plants grown after recombinant DNA process as seeds are not there? No, these plants will produce seeds because you are making the changes in a manner that you will be producing a line of their generation, and that's why you can use it further. About the economy prelims discussion not available, I had asked the team and I came to know that there has been some problem with the processing of the video, which is being re-uploaded now, so should be available very soon. All right, so let's move forward now. Apart from, so what we discussed is about how transgenic plants are made and how transgenic plants can be useful in various different ways, and we can use it for increasing the agricultural productivity, adding the nutritive value. There are so many ways in which we can make the use of agricultural application in transgenic plants. Now in case of other applications that we also have biofuels. We discussed about biofuels, and what we discussed is that primarily we are making the use of edible crop plants in biofuels. So that's why in all these cases you can practically say that biofuel is also an example of agricultural application of biotechnology, because you are using a plant, you are using an edible plant or a part of the plant to make your fuel. So that's why it is also something that can be counted as agricultural application of uh, biotechnology. Similarly, we have something called as biofortification. Now, biofortification becomes very, very crucial because this is where we are adding certain properties to the plants. Now, in this particular scenario, what when we took the example of golden rice, so golden rice is an example of biofortification. So biofortification is where you are adding to the nutritive value. All right, you are adding to the nutritive value of a particular crop, then or a particular, let's say, edible crop. Then in that case we say that it is uh, biofortification. So that's a development of micronutrient dense staple crop variant using the best traditional plant breeding practices and modern biotechnology will be called as biofortification. Now, micronutrient dense crops. What do we mean by micronutrients? Any idea? What do we mean by micronutrients? Yeah, the nutrients that are required in small amounts. Example wise, vitamins and minerals. Yes. So micronutrients are the nutrition or the nutrients required in small quantity. So that's why vitamins and minerals are examples. Vitamins and minerals are examples. So that's why whenever we are trying to specially add vitamins or minerals to any crop, then in that case we say that it is biofortification. And there are various practices of biofortification. We'll take those examples. Apart from that, sometimes we also make the use of supplementation instead of using it through a crop. Instead of increasing the nutritional value of the crop, sometimes supplementation also happens, where instead of adding it to the crop, what you're doing is that you are adding it to, let's say you are taking it through a medicine. Suppose vitamin D tablets or vitamin B12 tablets or bicosouls, etc. They are all supplements. These are all called as supplementation, wherever deficiencies have happened and medicines are being taken to counter those deficiencies. So that's why we see that these are things that we have to understand. So fortification is when you are adding the ingredient to in the food. So whether it is golden rice, or even in case of salt, when you have iodized salt, so that iodized salt also is an example of fortified food. So fortified food, or at the other end you have the ingredients where you can do it in the crops itself. Now, there is, when it comes to fortification, there is one issue that we also need to be aware of, and this is the issue of hidden hunger. Now, what hidden hunger according to WHO is when you are getting enough food but not enough nutrition. What I mean is, especially let's say for the poor countries where food supplies are being done externally, maybe some country is providing that food. So what happens is that they might get rice, they might get pulses, they might get all these. So they are getting good sources of carbohydrates, maybe good sources of proteins, but not a balanced diet. They're not getting enough of vitamins. They're not getting enough of the mineral requirements are not being fulfilled. So there are all these issues that they have, and hence this is where we say that it is hidden hunger, where although you are eating but you're not getting all the nutrients required. You are only eating something that is being given to you without looking at all the requirements of the nutrients that a person has. So that is called as hidden hunger, that when you are eating but you're not getting all the nutrients. Now, when it comes to biofortification, there are various different aspects of, there are various different crops that have been used over the time, and we have seen that in most of the cases we have been trying to enhance the level of one vitamin or the mineral. So overall, so let me just write here first that hidden hunger, so that you don't forget later, that when you are eating enough, or let's say the quality of food doesn't meet the nutritional requirements. All right, when the quality of food doesn't meet the nutritional requirements, this is called as hidden hunger. So that's why in, especially in the cases where the food is being exported or the food is being provided externally, we see that this is something that becomes a bigger challenge for those countries more than anyone else. So that's why WHO has been flagging these issues and saying that we need to find solutions to these kind of problems. Now, when it comes to biofortified foods, we have seen that there are various different types of biofortified foods that has been used overall. So whether it is cassava or maize or sweet potato or rice or millets, beans, everywhere we have seen that biofortified food has been used. So, and biofortification in all these cases have been different biofortification cases, and every mineral and, I mean, any mineral or any vitamin which is deficient, we have been trying to add to that. So that's why we have seen that in all these cases biofortification has been used. Now, in all these cases, when it comes to, let's say, biofortification, uh, we have seen that in cassava, for example, we have done vitamin A enhancements. All right, vitamin A enhancements have been done. Similarly, at the same time, we have also seen that iron and zinc enhancements have been done. So there have been cases of all these enhancements that we have tried in cassava. Similarly, in case of others, if you, for example, let's say in case of maize also we have done vitamin A enhancements. So that is also there. Then in case of sweet potato also, we have done vitamin A enhancements. So here also we have seen something similar. Then in case of uh, maize, there has been selenium example also, that we have increased the levels of selenium in maize, and that also is a form of biofortification that we have used. Similarly, when it comes to beans, then in case of beans, it has been largely about iron, that we have made we have fortified iron in case of beans. Then in case of millets, we have seen again that iron is something that we have done. So in case of millets, it has been iron and also calcium. Iron and calcium enhancements has been done here. So in all these cases, we have seen that in one way or the other. In case of rice and wheat, we have seen that vitamin A enhancements have been done, and also zinc enhancements have been done. So in all these cases, we have seen that there are various different ways in which biofortification has been carried out for all these various different food items. So overall, that's why we see that it's common that we have seen in all these scenarios. Now, apart from this, there are other ways. For example, in case of animal feed also, we have been making the use of we have been making the use of biotechnology to enhance the nutritional value of the food that the animals are getting. That instead of they having to eat a lot of food with limited quantity of food, how can we provide them the required nutrition? So that also has been a part of uh, biotechnology, and that's why we see that in animal feed it has been used. Then in pest resistant cultivars, where we took the example of bt cotton, so that we have done. Similarly, there are herbicide tolerant crops, which are the HT variant crops. In case of mustard, we have seen HT variant crops, for example, where we are making them herbicide tolerant. So herbicide tolerant is where they are actually making the use of uh, some microorganisms so that they can tolerate herbicides. Herbicides are used to kill the unwanted plant growth, or the weeds that are growing in the fields. So to kill those weeds, we are making the use of herbicides. So these herbicides many a times may also have a harmful effect on the crop. So that's why there are HT variant crops where the crop is not affected by the herbicides, but rather only the weeds are getting affected by the herbicides. So these are herbicide tolerant crops. Similarly, the use of biofertilizer, because in biofertilizer also you are using some animal or plant waste to enhance the nutrition in the crops. So that also can be counted as an application of agriculture in biotechnology, or application of biotechnology in agriculture. And then improvement in floriculture. So floriculture is basically about flowers. Now in flowers, what do you need? You would want the flowers to grow with good colors, good contrasts, maybe bigger flowers, maybe for with everything better fragrances or nice fragrances. And at the same time, you would also want longevity out of these flowers. So to enhance all these properties in flowers also, biotechnology is actively used. So it's not only the food, it's not only the food agriculture, but also we see that in cash crops also, in commercial agriculture also, we are making the use of, in commercial crops also, biotechnology is being used. For example, a couple of years we have seen that GM rubber also has been in the news, so which is also a commercial crop. So where we have created GM rubber in a manner that in areas where we are not able to grow rubber, we'll be able to do that. And especially in the northeastern states, two states specifically, in Assam and in Arunachal Pradesh, is where the target is. So there are all these cases where in one way or the other, biotechnology can become useful. Read this question. This was asked in 2011. I'm launching the poll for you. Or let me not launch the poll for this question. There is a reason why I'm not doing that. You tell me the answers in comments. What do you think? What is the answer? Mhm. As expected, a lot of C's and a lot of D's, and very rarely B and A, but B and A are not the answer. Let me discuss this question with you. Other than resistance to pests, what are the prospects for which genetically engineered plants have been created? First, to enable them to withstand drought. We have done it. We already have done it, and we have been doing it where the crops do not need a lot of water and they can also grow in water scarce areas. So that's why one is correct. Second is to increase their nutritive value of produce. We discussed this. Golden rice as an example and all the biofortified foods. So that's why increasing nutritive value of the produce is possible. So second is also correct. Third is to enable them to grow and do photosynthesis in spaceships and space stations. Very weird statement. Let's hold on. Fourth is to increase their shelf life. We discussed this as one of the desired properties. So 1, 2, and 4 definitely are correct. Now let's come to statement number 3. To enable them to grow and do photosynthesis in spaceships and space stations. Now this has been now done, that we have been able to grow plants in spaceships and in the international space station we have been able to do that. So now we have been able to do that, but this happened recently. This did not happen in 2011. So before 2011 or until 2011, statement number 3 was not correct. And look at the question. Let me just go back to the question and what the question says. Other than resistance to pests, what are the prospects for which genetically engineered plants have been created? Have been. Have been means what? That it should have happened by now, and then only you can mark a have been to be correct. It's not about the future. Another way to ask this question could have been can be created. Right? So then they could have said that what are the prospects for which genetically engineered plants can be created. Then when it comes to can be anything. So then in that case 1, 2, 3, 4 will be automatically correct without even thinking of a lot. But it says have been created. So have been created means that unless it has been done, you cannot mark it as correct. 1, 2, and 4 correct in even in the prospect of 2011, but statement number 3 not correct. So now that's why statement number 3 can be correct, can be wrong depending on when you are marking the answer. So the 2011 answer will be 1, 2, and 4, and 2024 answer will be 1, 2, 3, and 4, because now we have done it. So that's why I did not launch the poll. That if you are thinking from the perspective of when the question is asked, then 1, 2, and 4. And if you are looking at current perspective, then you can say 1, 2, 3, and 4. Sorry, this is repeated. Next is white biotechnology. Now, in case of white biotechnology, we are talking about industrial applications. Now, in industries also, there are various ways in which biotechnology becomes useful. So, for example, the fermentation process with which we produce alcohols, and then in that case, that can be used in various industries. For example, in biofuels, in making of bioethanol, the same fermentation process can be useful. Similarly, making of preservatives, making of some of the chemical reagents, we see that in all these cases biotechnology many a times is becoming very very useful. So when we are making the use of biotechnology in the industries, we are saying that it is white biotechnology. So microorganisms that can help in fermentation, so even in using in oil recovery. Now, in case of oil recovery, what we see is that the microorganisms will consume the oil, they will consume the hydrocarbons, and from these microorganisms, then oil can be extracted. So then they can help in oil recovery also. So they can help in enhancing oil recovery from the wells. Then they can also, as we discussed, can be used as preservatives, and as we discussed, they can be used also as biofuels. So there are various ways. There are some important important industrial applications also. For example, we have something called as biosorption. Now in case of biosorption, you can just think of it as two words coming together: biological absorption. So that's why the term biosorption. So biological absorption, where we are looking at a certain microorganism which can absorb. Absorb what? Absorb heavy elements, absorb toxins from any area which is affected, which is negatively impacted. So that's why all this can be counted as a part of biosorption. Now overall, so that's what we are saying is property of certain types of bacteria, which is inactive dead microbial biomass, to bind and concentrate heavy metals from even very dilute aqueous solution. So when we are trying to extract heavy metals or even toxins, for that matter, maybe from water, maybe from soil, so whenever we do that with the help of microorganisms, we will say that it is biosorption. So that is what the meaning of biosorption is. So overall, that's why it all helps us in industrial processes, and especially wherever you have contamination, wherever you have industrial contamination. Wherever we have industrial contamination, we can make the use of biosorption, and it will help us in cleaning up of these environments. So whether it is cleaning up water or cleaning up of soil. Then we have biomining. Now in case of biomining, we are talking about extraction of desired minerals. So between biosorption and biomining, process wise, microorganisms wise, it's not very different. All right. But in case of biosorption, we are trying to extract something which is harmful so that we can take it away from the environment. Whereas in case of biomining, we are looking for something which is useful, something that we can make the use of, whether it is in oil recovery, whether it is about metal extraction, with whatever it is, but in desired way. So something which is desirable for us. So that's why biomining. So in case of biomining, because we are doing the mining with the help of microorganisms or any organism for that matter, that's why biomining. It's not microorganism mining, it is biomining. So biomining meaning that when we are making the use of biological agents like animals or even plants or microorganisms to extract something, then in that case it is called as biomining. So in case of biomining, what we see is that this is where microorganisms can be made use of, or in case of phytomining, plants can be made use of. Now especially when it comes to microorganisms, just understand where they'll be used: in areas which have become dilute, in where you have concentrations which are pretty low for industrial methods to be carried on. Industrial methods can become costly here because you might use a lot of energy and yet not extract a lot. So hence what happens is that the value that you are generating is lesser than the consumption of money or the consumption of power that has happened here. So that's why here we see that this is where, in case of biomining, microorganisms will be used, especially in case of dilute environments where industrial methods can become costly. So microorganisms can be used, and then we have phytomining. Now in case of phytomining, we are saying that, just understand this, that wherever you come across this term phyto, right, phyto basically means plants. Phyto means plants. So simple terms, when we are saying that plants are being used, plants are being used for mining. Now how do we use plants for mining? The roots of the trees, the roots of the plants, they actually can go in the soil, and they can absorb the metal ions from the soil, and that is what can be used for extraction. So for example, let's say there is a dilute concentration of copper in any given environment. So then in that case, what happens is that copper can be used, that copper can be extracted from this environment. So copper extraction can be done with the help of the roots, and from the roots then copper can be extracted. We just burn up the roots many a times, and from the ashes we take copper out. So in this manner, we can use plants also for mining. So either microorganisms or plants can be used for mining purposes, and this will be called as bio mining. Then there is blue biotechnology. Now, in case of blue biotechnology, we are talking about the use in the aquatic environment, the use of biotechnology in the aquatic environment. Now, in the aquatic environment also, there are various ways in which we can make the use of biotechnology. Now, just understand how exactly we can make the use of biotechnology in the aquatic environments. Now, suppose that there is a oil spill has happened on the sea surface or on the ocean surface. Now, obviously, it leads to marine pollution, which is bad for the marine animals and the plants. So here we can make the use of biotechnology in cleaning up of the oil spill, where we make the use of bacteria, a set of 5 different bacteria, a formulation created out of 5 different bacteria, and this formulation will be just put to the place wherever oil spill has happened. Now what happens is that this bacteria will start eating up on that hydrocarbon. They'll start breaking down the hydrocarbon into carbon dioxide and water, carbon dioxide and moisture. So then the hydrocarbon will be broken down by the action of this bacterial formulation, and hence they would have just cleared the oil spill. And this bacterial formulation here is called as oil zapper, right? We use oil zapper, mixture of 5 different types of bacteria which feed on the hydrocarbon compounds, and oil zapper would convert hydrocarbons into carbon dioxide and water. So that's why we know that it can be done. So we can prevent oil spills by doing this. At the same time, we discussed about algae based biofuel, correct? We discussed about algae based biofuel. So that can be counted as blue biotechnology. Similarly, growth of underwater plants, underwater organisms, that can all be counted under blue biotechnology. So that's why anywhere, wherever you are looking at water related applications where biotechnologies are being applied to aquatic environments, then in that case it is called as blue biotechnology. All right. Then one of the very, very important areas in biotechnology is something called as bioremediation. Now, in case of bioremediation, we are talking about cleaning of the environment with the help of uh, organisms or living organisms. So, cleaning of the environment with the help of living organisms is called as bioremediation. Remediation is basically finding a remedy, correct? So, that's why we are talking about cleaning of the environment remediation. Now, there are two processes in bioremediation, one called as in situ bioremediation, the other one called as ex situ bioremediation. In situ bioremediation is in situ bioremediation. In situ, you might know the meaning. Can you tell me what is the meaning of in situ and what is the meaning of ex situ? Yes, at the site and away from the site. So in situ means at the site, and ex situ means away from the site. So in situ means at the site and ex situ means away from the site. So meaning that we are talking about when it comes to in situ bioremediation techniques, we are saying that wherever whatever contamination exists, if the contamination is being treated at the site of contamination, then in that case it is in situ bioremediation. And if you are taking the contamination away from the site and then treating it, then it is called as ex situ bioremediation. So that's the basic difference between in situ and ex situ bioremediation. So in situ bioremediation is when you are doing the treatment at the site of contamination, and ex situ bioremediation when you are taking the contamination away from the site and then you are doing the decontamination. So that is called as ex situ bioremediation. So that's the basic difference between in situ and ex situ. Now there are various processes that's there in as a part of in situ and ex situ. So let's try to understand that what is in situ and what is ex situ. So in case of in situ bioremediations, there are various different processes. So for example, we have something called as biosparging, bioventing, bioaugmentation, biostimulation, bioculture. There are all these things. All right. Now in case of all these methods, especially these two methods are very important: biosparging and bioventing. That what is biosparging and what is bioventing. Now in case of biosparging and bioventing, as a process these two are very close to one another. There isn't much difference between these two processes. Both basically will be making the use of indigenous microorganisms to treat the contamination. Now understand what's happening. All right. So suppose if we are talking about soil, so let's say this is the upper part of the soil, and below the soil layer you would have the water table. Typically this is what the case is. Now what we understand is that microorganisms are present. Microorganisms are present here in this layer of soil, correct? And in the water bodies also microorganisms are present, and these microorganisms are helping in bioremediation. I mean, naturally itself they are helping in decomposition. So that's why it's not that they cannot be used. They can actually be used in cre- uh, cleaning of this environment. Now overall what happens here is that although natural cleaning will happen, but this natural cleaning takes time. Natural cleaning takes a lot of time. So that's why in this particular scenario especially, we cannot really rely on microorganisms a lot, because although naturally they would decontaminate the area, but we want faster results. And for getting these faster results, what we do is that these microorganisms are given extra nutrition. So we inject extra nutrition to the soil and water, and by injecting that extra nutrition, these microorganisms get that extra nutrition, they get that energy, and hence they start working faster. So to make them work faster, what we make the use of is that we add extra nutrients. We are saying that the nutrition is being added, microorganism was already present. All right, microorganism was already present. So that is basically where we see these two methods of biosparging and bioventing, where biosparging is used in the saturated zone, whereas bioventing is used in the unsaturated zone. All right. So unsaturated and saturated, understand this. Now this upper part is called as the unsaturated zone. So when nutrition is being introduced here, when oxygen and nutrition is being introduced here, this is the unsaturated zone. And when oxygen and nutrition is inserted here, this is the saturated zone. All right. So I'm just writing it here. This is the unsaturated zone. And the one which is saturated with water is called as the saturated zone. So unsaturated zone and saturated zone. And that's why just remember that in unsaturated zone, we carry out bioventing, and in the saturated zone we carry out biosparging. So what exactly are we doing in bioventing and biosparging? In case of bioventing and biosparging, we are injecting the nutrients. Now, injecting the nutrients to the unsaturated zone will be easier. So that's why it can be done at lower pressure. It can be done at lower pressure. It's easier to inject the nutrition to the upper part of the soil. But when we are going to the saturated zone, we have to do it at high pressure, because injecting of oxygen and nutrition to this layer will not be that easy. So that's why biosparging is done at high pressure. Injecting oxygen and nutrition at high uh, pressure into the saturated zone is called as biosparging, and injecting oxygen and nutrition at low pressure in the unsaturated zone is called as bioventing. So that's the basic difference between biosparging and bioventing. In both the cases, you are injecting the nutrition to the already existing microorganisms, all right, and trying to enhance their energy so that they can actively start working. So that's precisely what we are trying in this particular scenario. So that's why air or oxygen and nutrition are injected at high pressure to increase the biological activity of the indigenous microorganisms and to enhance their decomposition activity. So this is what we do in case of biosparging, and in case of bioventing, we are using low pressure, right? We are using low pressure, and this can happen at low pressure, where we are inducing oxygen and nutrition in the unsaturated zone, and that is how we see that these two processes are pretty similar to each other. Apart from that, what we discussed is that there are other processes also, that you have bio augmentation also as a process. So in case of bio augmentation, this is where you bring microorganisms to the site, all right. This is where microorganisms brought to the site of contamination. So microorganisms are brought to the site of the contamination. Example we just discussed, oil zapper. So in case of oil zapper, what are we doing? That we are bringing this bacterial formulation to wherever the oil spills have happened or oily sludge is present. So that will be brought there, and it will take care of the problem at the site itself. So that's why it is in situ, and it is called as bio augmentation. So that's why overall we see that there are different ways in which they can be utilized. So so that's why there are various different methods, and we have just named a few. There are other methods also that are used when it comes to uh, in situ bioremediation techniques. So apart from that, when we come to the ex situ bioremediation, again in ex situ bioremediation also there are various techniques that we are using. For example, in case of land farming, so in case of land farming, what we do is that we take the contaminated soil somewhere, and then periodically there will be tillage of this land that will be done. Tillage of the land will also mean that the microorganisms are being woken up, that they have become very dormant and they do not have a lot of nutrition, etc. So they just have gone to sleep. So this is where they are stimulated with the help of plowing or with the help of uh, the technique of tillage. So that's why the soil will be periodically tilled. The soil will be periodically tilled in soil will be periodically tilled to stimulate the growth of the microorganisms for degradation of the organic waste. So you take the soil somewhere, and then the tillage of the soil will be periodically done there, so that we can enhance the quality of the soil by waking up the bacteria, waking up the microorganisms in a manner, you can say. Now, one technique here which is very, very useful is the technique of biopile. Now, biopile, what we do is that, as it says here, it's a technique which is a hybrid of land composting and land farming techniques. Now, in this particular scenario, what we do is that excavated soil are spread over prepared bed, and as you can see in the picture here, and then after that it will be enclosed for treatment. And then here we take care of each and every aspect of the soil, that what kind of soil do we want to prepare from here. So what should be the moisture content of the soil, what should be the heat content of the soil, what are the nutrients required, oxygen presence, what should be the pH level. So each and everything will be taken care of, and everything will be controlled here in case of biopile. So these will become the biopiles. These, as you see here, these are the biopiles where you have treated it and you then keep it for a longer time. Also, we will introduce certain decomposers, or let's say earthworms, etc., and we'll try to stimulate the soil better. We'll try to add nutrition to the soil. So typically it can take up to 3 to 6 months of time for this soil preparation. So overall it will become a method that can be utilized in all these cases, especially in agriculture. Then, apart from that, there are other techniques like phytoremediation. Now, as we just discussed, that phyto means plants. So when we are making the use of plants for any kind of decontamination effort, then we can say that it is phytoremediation. Now, in case of phytoremediation, uh, we know that plants, you can say, by their nature, because of how photosynthesis happens, they clean up the air, because they will take in carbon dioxide and they'll give out oxygen. So overall, you can say that plants anyway are helping in bioremediation. But beyond that, also we see that plants are useful. For example, the neem plant, known for all its properties, that whether its flowers, or whether it is the seed, or the bark, or the leaf, every part is useful in one way or the other. And that's why we see that there are a lot of applications that we have of neem plant, that it will help in cleaning up the air. It also has antimicrobial properties, so it will also help in decontamination from the microorganisms. So there are various ways in which it can become pretty useful. So that's why the use of plants to remove contamination from soil and water is called as phytoremediation. Now, there is also something called as phytoextraction. Now, in case of phytoextraction, you can say that this is where the plants are extracting the toxins out of the soil. So that's why phytoextraction, it's an extraction technique. So here what we see is that the roots of the plants will just absorb all the toxins. It will absorb all the heavy metals, etc., that would have been built in the soil. So in that case, we'll see that the roots of the soil here, they will be useful, they will be helpful in ensuring that the soil quality can be enhanced by taking away all the heavy metals and other toxins which might be present in the soil. So that's why this is where the plants will remove the dangerous elements or the compounds from soil and water. So this is where you have a process called as phytoextraction. Then there is also something called as mycoremediation. Now, in case of mycoremediation, we are talking about the use of fungus, or what you call in plural as fungi or fungi, or whatever you want to call it. As long as you are writing the right spelling, nothing else will matter. So that's why a safer word to use here is fungus. So the use of fungus in case of is called as mycoremediation. Now, in case of mycoremediation, we see that especially the term myco itself, just remember this, that the term myco itself signifies fungus. So that's why when we are making the use of fungus, we are saying that it is mycoremediation. Like nitrogen fixation, that we know that there are fungi which will help in enhancing the amount of nitrogen that is present, or nitrates that are present in the soil. So they will enhance the soil quality in that sense. So that's why they are also helping in they are also helping in bioremediation. So that's why we see that there are various different examples. I mean, there are so many of them. We know that all these are used in one way or the other. There's this one question also that has been asked from here. Just read this question. And just give me 1 minute, I'll be back. I need to fill water. My water bottle is empty. Yeah, I'm launching the poll for you. Please mark your answers. Hmm. So there's a tough competition between C and D, I see. So question says, in the context of solving pollution problems, what is or are the advantage or advantages of bioremediation technique? First is, it is a technique for cleaning up pollution by enhancing the same biodegradation process that occurs in nature. So this is fine. This is what we anyway discussed, that where we are talking about how we can enhance the activities. In case of biosparging and bioventing also we discussed the same thing, that we are trying to enhance the activities more than anything. Then any contaminant with heavy metals such as cadmium and lead can be readily and completely treated by bioremediation using microorganisms. Now see, there are two things here. All right, first the sentence itself. Any contaminant such as with heavy metals such as cadmium and lead can be readily and completely be treated by bioremediation using microorganisms. These are all extreme words. These are all words which we can call as extreme words. Now, presence of extreme words largely means that you are making an absolute statement, right? Any contaminant completely can be treated, right? So especially when it comes to these absolute statements, unless you are 100% correct, you are 100% sure that this is a right statement, there are huge chances that these statements are not correct. And this is what you will find, that in 9 out of 10 of these statements, if there are extreme words used, chances are that these are wrong statements. And this is precisely what's happening here also, that the sentence itself is a very absolute statement and hence it's wrong. But why it is also wrong, just understand this, that when it comes to heavy metals, For microorganisms, it's very difficult to break down the heavy metals, so they would either absorb it or will try to do something out of it. But complete treatment with microorganisms becomes very difficult. We largely use plants in these cases. We largely use plants. Phytoremediation is used largely when it comes to heavy metals or contaminants which are made of heavy metals. So that's why it's not microorganisms, but largely it is about plants. So that's why this is wrong. But anyway, as I told you, that the statement itself is very absolute in its nature, and hence it is also wrong. Genetic engineering can be used to create microorganisms specifically designed for bioremediation. This is correct. We discussed about oil zapper, and oil zapper is a basic example. So that's why correct answer will be 1 and 3 only. Now, extreme words. Do you understand the meaning of extreme words? Can you give me examples of extreme words? Yes, Dinesh, you are correct. Any, all, always, every, never. I'll write it. Many of you have written the examples. So any, all, every, always, as you have here, completely, never, none. So all these, you can say that these are all extreme statements. Only, this is also an extreme. So that's why all these, whenever you see anything that looks like very extreme, you can always mark it as, I mean, you can always at least think twice before you are marking something. So that's why they become very very crucial in marking your answers. And you'll find that many of the previous year prelims questions also, if you notice, you'll find that wherever they make absolute statements, they largely are wrong in making those absolute statements. All right. I think this is something that you'll also learn in reading comprehension, and how in reading comprehension we also come across these terms. Another question. Recently, oil zapper was in news. What is it? Very simple. We just discussed it. I'm launching the poll for you. Wonderful. So 80% of you have given the correct answer, which is A. It is an eco-friendly technology for the remediation of oily sludge and oil spills. Others are not related to oil spills. We discussed that oil zapper is used in oil spills, so that's why others are not correct. Now, before moving on to the next topic, if you have any questions. When oil zapper have cleared oil spillage, then what do we do with the oil zapper removed or left there? We just leave it there because these are microorganisms. Doesn't do not make any difference uh, in that environment. So nothing to be done for them. If bacteria breaks hydrocarbons and will get H2O and CO2, then won't these increase carbon dioxide component will affect aquatic life and environment at large? They wouldn't, because it's not a huge amount of carbon dioxide that is being emitted from here. And also, it is not something which is happening all of a sudden. All right. So it's, if let's say for example, if it was happening quickly, that all of a sudden there was a sudden burst of carbon dioxide, then it will be a problem. But this is a small breakdown, this is a very slow breakdown rather. So that's why in this particular scenario, we cannot really say that uh, it will have any offset in the environment. Biosparging. So in case of biosparging, what we are saying is that in biosparging, we are making the use of microorganisms which were already present in that area, and then we are injecting nutrition to them. We are giving them nutrition so that they can gain energy and their activities can be enhanced. All these topics are given in 10th by biotechnology. No, I don't think in NCRT books it's there. In spaceships, crops have been grown, but photosynthesis still not achieved. No, in International Space Station, we have done. There was a paper that was published on it. Ionized salt is GM food. No, it is not GM food, because genetic modification is being not done in iodized salt. You're just adding iodine chemically. It's not biofortification. It is just fortification. All right. So it is not GM crop. Any side effects in bioremediation technique? Uh, Depends on what you're using for bioremediation. If let's say some chemicals are being used, or let's say something that can enhance the chemicals can be are being used, then it can create some problems. What is the meaning of saturated and unsaturated? So when we say saturated, the meaning of saturated is that where the concentration of water is almost 100%. So when you have very high concentration of water, like the water bodies itself, or let's say even let's say in soil where you have a lot of moisture, and it cannot intake more moisture, then in that case you can say that it is saturated. Difference between bioremediation and phytoremediation? No, it's not a difference, but rather you can say phytoremediation is a process as a part of bioremediation. Bioremediation means any organism, and phytoremediation means specifically plants. That's the difference. Antimicrobial means, the meaning of antimicrobial is when you're doing something against microbes, so that's why antimicrobial. Dilute environment means whenever you do not have high concentration of something, then you will say dilute environments. How much time bioremediation takes to complete depends on the process. Every process takes different amount of time. Difference between fortification and biofortification, as I just told you, that fortification is any enhancement in nutrition, like iodized salt being an example, where iodine is being added to the salt to fortify salt. But bioremediation is when you're using a plant, and when you're using something based out of that plant, then it is bioremediation. So anything, I mean, not plant, rather let's say any organism for that matter. So when you're using any biological resource, then it is biological or bioremediation. So sorry, biofortification. Otherwise it is just fortification. How can we fortify rice? I mean, the same process is used. I mean, when we are trying to enhance it, either hybridization, hybrid varieties can be created sometimes, or we can also make the use of genetic modification with the help of recombinant DNA technology. So bioremediation, just understand, very simple. For those of you who have asked the definition again, it's very simple that we are using an organism. You just take any organism and you are using it for something. Let's say neem plant. We know neem plant for its medicinal properties, right? So we know that it helps in cleaning the environment. It has it can be used against germs, against diseases. So that's why bioremediation. So bioremediation, anything which is cleaning up the environment, any organism that is cleaning up the environment. That even, let's say, uh, many of the food varieties also, like spinach, for example. Spinach can remove iron from soil. So that is also bioremediation, that it is able to remove iron from soil. Similarly, I think maize I think can remove lead from the soil. So it will be able to also clean up the soil. So in this manner, all these things, wherever any organism is being used for cleaning up the environment, is bioremediation. Hmm. So the next topic for us is cloning. Now, when it comes to cloning, the meaning of cloning is what? That this is where we are producing an exact replica. Cloning means this, right? In general, when you say that you're cloning something, cloning means that you're producing an exact replica. So producing an exact replica means what? That exact copy is being produced, a photocopy is being produced in a manner. Now, how can we produce an exact replica of an animal? In the movies you might have seen that there is a person who would go into a box, and from in another box another person comes out, and thus cloning is done. If for those of you who might have seen Shaktiman, there is an episode when Shaktiman's clone is produced. So similarly, there are many English films also where you'll see that they have made the use of clones like this. They'll just show you that somebody has gone in a box, and then from there on another person comes from another box. So in that case, that is cloning. But unfortunately, in real life, things are not that simple, that you cannot just push somebody in one box and another person comes out of the other box. In case of cloning in animals, what we do is, for creating an exact replica, we use this entire process. So what we say is that cloning is a process of asexual reproduction, in which the offspring or the progeny is an exact replica of the single parent who has contributed to the genetic material. So this means what? First, we are saying that it's a process of asexual reproduction, meaning, now reproduction, you might know that there are two types of reproduction, one called as sexual, another one asexual. Sexual is where you have a male and a female involved, that there will be genetic mixing between the male and the female, and the offspring will be a mix of the genes from the male and the female. Whereas in case of asexual reproduction, there is only one single parent, and from that one single parent reproduction is happening. For example, cell division. In many smaller organisms, with just cell division, one organism divides into two. First, the nucleus starts to divide into two nuclei, and as it divides into two nuclei, it separates out. So that basically is how we look at uh, the process of cell division being asexual reproduction. Similarly, there is a process of budding, where a bud will be formed on the organism. The bud grows and then it detaches from that organism and becomes an organism in itself. So then in that case, what we see is that in this scenario also, this is also asexual reproduction. Now in case of cloning also, we are saying that it is asexual reproduction, mainly because of the fact that here also only one parent is involved. All right, only one parent is involved, and you are replicating the cells of that one parent to produce a new organism or produce a new animal in that particular sense. So this is what cloning is, that where offspring or the progeny is an exact replica of the single parent who has contributed to the genetic material. Now overall, when it comes to cloning as a process is where we see that the entire organism will come out of the cell that we have taken from that animal. Now, genetic modification in animals in general is not as easy as it has been in plants. Now, even in plants, it's not easy. But when we look in relative terms, I would say that in case of plants, carrying out genetic modification is easier as compared to in animals. The animal genetic structure is far more complex, and that's why doing any genetic modification or any biotechnological process in case of animals is extremely difficult. And that's precisely what we see with cloning also, that the success in cloning hasn't been that great. Although we have established the process, we have been using the process, but it's not that easy to make the use of cloning. So when it comes to cloning, for example, this one example that you see right here, this is the first successful example of cloning. This is a sheep, cloning of a sheep called as Dolly, which was done in the year 1997. Now, even this first success that we got in cloning, do you know how many attempts it took for us to be successful? Any idea? A trivia question, a KBC question for you. How many attempts did it take for us to be successful in cloning? 5, 10, 50, 100. I'm getting all these. Janta, I'll look into your issue, what exactly your issue. If you can just send your email to me also, it will help. Yeah, Ziva, you are correct. One correct answer. Dipender, you are correct. So it actually took 277 attempts. So for those of you who said 5 attempts or 10 attempts, it took 277 attempts for us to be successful. How many attempts do you get at success? I'll not go there. So uh, so that tells us how difficult or how unstable this process has been, that it took a total of 277 attempts for us to be successful in getting cloning right. And even then, even after this success, it's not that the success has continued, uh, and we have been able to stabilize it. So if you look at the Indian examples with animal cloning, the first Indian example was of Samrupa back in 2009, and this was the world's first cloned buffalo. But it did not live for very long. It did not live for very long, and it just after 5 days after its birth, it succumbed to lung infections and it died. Then the second case was of Garima, again in 2009. It lived a little longer, lived for almost 2 years, and then again, because of heart failure, 2 years later, it did not live longer than that. And then in 2016, it was Gaurav. This was the third case of uh, buffalo cloning in India, and this is where we see that this has been better. Apart from that, there have been some recent efforts in cloning, where in 2020 and last year in 2023, these clonings have been done. Now, in 2023, this was the case of 2023 when the first cloning of a cow was done. Now, this basically has been done for a cow species in India called as the Gir cow, and the name of this has been given as Ganga. Now, for the first time, what we are doing is, I mean, in 2023, why this cow cloning was done. Basically, this is where for the first time we are also now doing the cloning of the indigenous variants. And the basic understanding here is that the indigenous species are better at surviving in India because they are used to these tropical conditions. Their genetics have been made for these tropical conditions, the kinds of temperatures and the moistures that India has. So that's why the other variants, the other species from, let's say, the European variants, etc., they would not be as good as in making themselves suitable in the tropical climates as the Indian variants would be. And now we want to enhance the milk production. So in enhancing the milk production is where the first step is being seen as this particular cloning. So this is basically related to enhancing the milk production in India. So this is a cloning effort. Before this, we see that in 2020 also some cloning efforts were done. So there were 7 clones of M29 Murrah bull, and then Hisar Gaurav also was recloned. So there was all these examples where we have seen it, where we have seen that all these cloning have been done. So overall we see that there are all these things, and that's where this is where cloning is advantageous. This is where we are looking at the process of cloning being advantageous, because wherever we want to make the use of animals proactively, this is where cloning can be seen as an example. So whether it is milk production, or whether it is in livestock, whether it is in farming, in agriculture, in all these scenarios we see that animal cloning can be used. The ones which are, let's say, genetically looking to be superior, the species who are good, who are who have a better resistance against diseases, we would want to clone all of them. We'll want to clone all of them so that we can build in better species or stronger species altogether for our use. So that is where we are looking at the process of animal cloning. Now, as far as human cloning is concerned, human cloning is banned. Human cloning is not allowed, and we'll discuss about the reasons for not allowing human cloning uh, probably in the next class. One thing before we end the class for today, the process of cloning, that how exactly is the process of cloning carried out. Now, process of cloning starts with a somatic cell. All right, it starts with a somatic cell. Now, somatic cell is any cell in our body which is not reproductive in nature. For humans, let's say there are two reproductive cells, the sperm cells in the males and the egg cells in the females. So these two are the reproductive cells. Apart from these reproductive cells, all the other cells are called as somatic cells. So somatic cells are the non-reproductive cells in the body. So first we isolate the somatic cells. All right, first we will isolate the somatic cells, and from these somatic cells first we take out the nucleus. The nucleus of the somatic cell will be taken out, and this nucleus will be placed inside another egg cell. So something like this happens, that first we will isolate the somatic cells, and from the somatic cells we will isolate the nucleus out. We will take out the nucleus, and this nucleus will be kept inside, will be placed inside an enucleated egg cell, meaning the egg cell from which nucleus has been removed. So we remove the nucleus of the egg cell, and we place the nucleus of the somatic cell inside the egg cell. So that's when now you have the egg cell with the somatic nucleus, and then activation of this is done with the help of electric shock, with the help of chemicals. We produce a bunch of cells, we replicate the cells, we produce a bunch of cells, which is called as blastocyst. This is called as blastocyst. So blastocyst is nothing but a bunch of cells which have been produced here. So now with this, we ultimately what we do is that after this blastocyst is produced, either this goes inside the host mother, inside the uterus of the host mother, where the embryonic development would start into an animal. So that will be called as reproductive cloning. All right, that will be called as reproductive cloning. And the second case is of therapeutic cloning. In case of therapeutic cloning, we do not want the birth of a new individual or a birth of a new animal. Instead, we want to harvest these cells, use these cells in stem cell therapy. We'll discuss that later, that how in stem cells this is being used. So that is, there are two things that can be done. Either here you will have reproductive cloning or therapeutic cloning. Reproductive cloning, when you want the birth of a new animal out of this cloning process, and therapeutic cloning, when you want to use all these cells for therapeutic purposes, then in that case it will be called as therapeutic cloning. So this is where we look at these two types of cloning. Now one thing here is that we are removing the nucleus from the egg cell, and we are placing the somatic nucleus inside this egg cell. Now why do we do this? What is the reason behind removing the nucleus and adding the nucleus? The basic reason here is that we discussed that all the genes which have specific characteristics of an individual are there inside the nucleus. So if the nucleus is removed, let's say when the removal of this nucleus happens, it means that all the specific characteristics that that egg cell had have been removed, and now we are placing the somatic nucleus inside the enucleated cell, meaning that now this egg cell will have all the individual properties, all the specific properties of the somatic cell, of where the somatic cell has come from. So that's where we see that all these, it's like doing a brain transplant. In simple words, you can say that it's like doing a brain transplant, that I have removed my brain and I have replaced it with Einstein's brain, so that I become much better in physics than I am. So, so that is basically what we are doing in case of cloning, that we are taking the somatic cell, and then we are taking the nucleus from the somatic cell and putting it inside the egg cell. So this is where we see that these are the two kinds of cloning, one called as reproductive cloning and other one called as therapeutic cloning. So process of cloning again starts with somatic cell, where the nucleus will be transferred into an enucleated cell, meaning the egg cell where the nucleus has been removed. And then the egg cell so obtained, then it is stimulated with the help of electric shocks and chemicals. And this is where we'll see that a single cell will become a ball of mass with around 150 unspecialized cells, which is called as blastocyst. And once this is done, this is inserted inside the uterus of a surrogate mother, where the uh, process of embryonic development can start from there on. Or if we don't need it, then in that case it is therapeutic cloning. So this is what the process of cloning is. All right, this is what the process of cloning is in this particular scenario. Now, there are other aspects of cloning and the problems associated with cloning, but I don't think we have enough time to start that discussion. We'll continue with the discussion of cloning in tomorrow's class. So, we'll end this part of biotechnology tomorrow, and then we'll start with our next portion, that is about nanotechnology. So, that will be the next chapter that will begin in tomorrow's class. So, if you have any questions from whatever we discussed in the last 20 odd minutes. What is three parents baby? We'll discuss that in tomorrow's class. It is there as a part of your chapter, so we'll discuss it. Designer baby and cloning, the same thing? Not exactly. Again, something that is for discussion in tomorrow's class. Here we are using egg cells nucleus. How is it asexual? It is asexual because uh, the fertilization that happens between the male and the female cells, that is not happening. We are skipping the process of fertilization. When the fertilization would have happened between the male cell and the female cell, then in that case we could have called it as sexual reproduction. Only the involvement of egg cell does not mean that it is sexual reproduction. There should have been the male counterpart as well. Any name of somatic cells? No, all the cells in the body are somatic cells. For example, let's say the nerve cells, skin cells, all these are somatic cells. Is cloning ethical? There are issues with it. We'll discuss that later tomorrow. Which traits come into the clone baby? Any trait that was there in the somatic cells, wherever we have taken it from, that somatic cells traits will be transferred to the child. RNA will be different for every individual or the same? It will remain the same for them. Leaderboard. Leaderboard. Yeah, this is the leaderboard for today. Congratulations to all of you with all the correct answers. Therapeutic cloning. Therapeutic cloning is when we are using cloning for medical purposes, for some kind of medical treatment, stem cell therapy. Then in that case, it is therapeutic cloning. Hmm. So a lot of you getting all the correct answers. Congratulations to everyone. For those of you who could not, I hope you understood where you went wrong. Sham, you'll have to practice. I mean, you'll have to just go through all that information because there are a lot of information that we have in this chapter. So just go through them, revise them. Any difficulty if you find didn't understand something, just ask it in the question in the class. About extra classes, I'll have to check with the schedule with the team, and we will announce the extra classes. Whatever we have missed, accordingly, we'll do it. Genome sequencing. Genome sequencing is where we are trying to find the sequence of the entire genome. That we know that there are so many genes in the body. Now, what is the sequence of those genes is what we are trying to figure out as a part of the Human Genome Project. So that is what genome sequencing is. That we know that there are 20,000 genes, but how are they arranged? So that arrangement is finding that arrangement is genome sequencing. How do we get the egg cell to plant the nucleus? Basically, it can be taken from any female body or anyone who is a surrogate. The tomato that went missing in International Space Station recently, was it genetically engineered one? Yes, it was a hybrid variety. Yes. Can we change characters during this? No, the characters will be retained. Cloned humans will also be exactly same as host humans because the genes will be the same. So that, but the genes also evolve. It's not that the genes always remain the same since your birth to your death. It keeps on evolving and changing. So those variations will come in later stage, but initially it will all be the same. Are somatic and stem cell the same? No. Stem cells are completely different. We'll discuss that later. As human cloning is banned, so how we producing clone baby? We are not. It's banned. We are not producing human clones. In recombinant DNA technology is 100% successful. Uh, Success, I mean, largely we have been successful, 100% to never. Can we recreate extinct species? We actually can. We'll discuss that tomorrow. Will cloned animals have ability for sexual reproduction? There have been complications that we have seen there. So uh, the answer would be not entirely. Nishan, thank you for your advice. I'll ensure it. Somatic cell taken from mother itself? No, not necessarily. You can take it from any individual. The legality of cloning, we'll discuss that tomorrow. So a lot of questions that you have asked, these are questions that actually I'm going to discuss that tomorrow later. So just wait for it. Surrogacy and cloning are different. Again, something that we'll discuss tomorrow. Yeah, scheduling of the class, we'll try to do it as early as possible, but I don't know when the scheduling can be done, but we'll try to uh, do it in this month itself whenever the time permits. All right, so let's end the session here. Please go back, revise it. There are a lot of terms that we have come across in this chapter. So your revision will be required so that you don't forget anything, and we'll continue from here. So we'll discuss about cloning, stem cells, DNA fingerprinting, three parents baby, GM mustard, all these things we'll discuss in tomorrow's class. And then we'll begin with our next chapter, which is nanotechnology. So these are the things that we'll discuss in tomorrow's class. Thank you.